Good morning, everyone. Today is the day 19 of 4-week technology-based entrepreneurship development program that is focused on bioentrepreneurship. It is sponsored by the Department of Science and Technology, Ministry of Science and Technology, Government of India, New Delhi. Today we have 3 keynote speakers, Ms. Bhavna Pandya ma'am. She will talk about the overview of bioentrepreneurship, their scopes, and Dr.Surajit Debnath, who will conduct a practical session on automation of enterprises goals using AI tools that is focused on biomedical entrepreneurship. And we have Mr. Vivek Singh, who will deliver the talk on regulatory requirement for the bio startup. So let's start with our session. Our speaker has joined with us. Good morning, ma'am.
Hi, good morning.
We would like to thankful to you, ma'am, for accepting our invitation. And Bhavna ma'am is the head of Bioriidl, India's first DIY biolab and biotech business incubator based in Mumbai. She is the innovation catalyst at riidl, Sumaya Vidya Vihar, and chief organizer of Darwin Conference. She is one of the successful community leaders who are paving a path for the biologists who seek lab facilities, guidance, and opportunity to create their own projects and startups. She has raised about $500,000 funding for the lab from the Bioriidl, Department of Biotechnology, Government of India. She is mentoring approximately 28 startups in food, agriculture, healthcare, industrial biotechnology, and synthetic biology, as well as other technology-based startups. She has 1 patent, 2 research papers published in the reputed journals, and also she has successfully graduated Bioacademy 2016 from the 5 Foundation run by Center for Atom and MIT Media Lab, and is the only bioacademic local instructor in India. She has initiated various projects in biomedical science and has also practices the research with the designers through one of her initiative called Bio Fashion Show, which has helped fashion designer to adopt the biomaterial in the form of clothing and accessory. So we are very grateful to have you as a speaker, ma'am. Thank you so much for accepting our invitation. So over to you, ma'am.
Thank you so much. So I'll quickly start sharing my screen and then I'll take you all to the...
Yes, ma'am, you can share your screen. Any difficulty?
Just a minute. I'm trying to share the screen. There's some issue. Is my screen visible?
Yes, ma'am. It's visible.
Okay. Hello, everyone. Good morning. So today I'm going to talk about the bio-entrepreneurship, right? So when we say bio-entrepreneurship, what exactly it means? How is it different than the normal entrepreneurship? So I would say there's not much difference apart from the risk associated with bio-entrepreneurship. So in general entrepreneurship, you have your ideation, you do a lot of you build the technology, launch it in the market, and then there are some or the other modification that you do in your technology. But when it comes to bio startups, it becomes very risky. It has a lot of risk associated with different kind of technologies involved. So, for example, if you are developing a drug, right, and you want that drug to go through a lot of stages, and if it fails even at one stage, your entire technology will go in vain, right? You won't be able to carry it forward because you won't get any approval further. So that's how it becomes a little risky compared to the normal other technology startups. That's why the new term has been coined for bio entrepreneurs, which is bioentrepreneurship. So I'm Bhavna Pandya. I lead this Bioriidl, which is India's first do-it-yourself biology lab, which was started with an aim of building a platform for biologists across India who can come and experiment their ideas. So people who doesn't have any lab or academics or even industries who doesn't allow such external projects, this is the platform where you can come and build your project. And once you have the project ready, you can get into the incubation phase, right? I'll tell you what all phases are involved in the bioentrepreneurship journey. So let's talk about Indian bioeconomy. Yeah. So Indian bioeconomy is growing very rapidly in last 5 years, and during the COVID, I think it has entire country realized how important the bioeconomy is for India, because that time everything was shut, but only the bio field of pharma companies were open, and they were so burdened with a lot of requirements that they had to double their production capacity. Right. So Indian bioeconomy is growing at a rate of around 14.15%. And from 2015 to 2020, it has doubled from 30.2 billion to 70.2 billion in 5 years. And as per the prediction that it is going to grow till 150 billion in by 2025. So here a lot of industries contribute in the bioeconomy. It's not just pharma, but there are industrial biotechnology companies, startups, and various other agricultural company or diagnostic companies. A lot of these companies are contributing in the in making this Indian bioeconomy stronger day by day. And here you can see Indian biotech or life sciences startup scenario. So India has been historically known for its IT and pharma companies, but now now it is also forced to renown as a global biotechnology market. So India is right now boasting around 5,000 biotech companies, with more than 4,000 being startups. Right, only thousands are the companies who are established or are registered as LLP, but out of 5,000, there are 4,000 startups involved in this ecosystem, and this count is expected to reach 10,000 by 2024. So India already had this huge base for IT talent, for hardware talent, for manufacturing pharma and vaccine drugs. But now, due to various government schemes, government is also trying to boost the entire life sciences ecosystem in India. We already know how important it is to have these life sciences companies growing in the country itself, so we become independent of any other country. And one of that very good example is Bharat Biotech, right? Bharat Biotech developed the first vaccine in India, which actually saved a lot of lives during COVID. So otherwise, we had to put a lot of money in importing those vaccines from different countries, and that time it was already burdened. The companies were already burdened outside with requirements, and our requirement would have not been fulfilled, or we would have been charged a lot for that. But thanks to Bharat Biotech, so they have developed that vaccine, and they actually fulfilled the need of the country. So that this particular area is that important for the country, right? Now, if you see the market breakup by revenues for this is financial year 2016, So biopharma is contributing a lot in the market, which is approximately 64%. Then there's bioservices. So when we say bioservices, it means providing consultancy research to various companies and industries. Then bioagri, which is again agricultural field, which is 14%. 3% is biotech, core biotech industry, and 1% is bioinformatics. Now this is one of the Make in India programs started by government. So there are a lot of part associated with it. So when we say global competitiveness of India, India is ranking #1 in ease of doing business in South Asia. It is also #1 in support of DPT, BCG, and measles vaccines quality globally. Then we are the second highest US FDA approved manufacturing plants outside the US. Then 3%, we share 3% in the global biotechnology industry. Then we are third largest biotech destination in Asia Pacific region, and we rank 52nd rank in Global Innovation Index. Now we are also enabling a lot of policies at center and state level. So we are expecting to grow by $150 billion by economy by 2024 to 25. Government has revised various policies for ease of doing a business for biotech industries. So that started with regulated policy at national and state level. So National Biotechnology Development Strategy, if you see that there are a lot of policies listed and modified. Then there's FDI policy launched in 2017. Medical devices rules were regulated in 2017. National Policy on Biofuels 2018, guidelines for evaluation of nanopharmaceutical in 2019. And then there's national guidelines for gene therapy, product development and clinical trials, which was developed in 2019. And then guidelines are available on biosimilar and biologics, which was again modified and developed in 2016. Apart from developing these regulatory policies, government also launched skill development policies. One is Pradhan Mantri Kaushal Vikas Yojana guidelines, which is again, you can find it on the website. Scheme for facilitating startup intellectual property protection, SIPP, which is 2017. So we already know that if you're filing a company, an IP as a startup, your application is fast tracked. So this government did to enable them, this use of IP to commercialize their technology as soon as possible. Otherwise, the normal duration for getting the IP granted is 7 years. Then a National Innovation and Startup Policy 2019 for students and faculty in the center. So if there are policies available for startups and faculties who would want to do startup in their university. So there are various schemes also available. One is Sitare. So if you look at the BIRAC website, BIRAC, I'll tell you about BIRAC. So BIRAC is an arm of DBT, which is focusing only on developing the bio-entrepreneurship in India. So if you look at their website, there will be a lot of grants and funds available for doing the biotech products or following your ambition in developing your company in biotech space. So they have grants right from the ideation stage to the late and maturity stage also. Even if you are an individual not sure, whether you want to do a startup or you don't want to do a startup and you don't have funds, you can also check the EUA fellowship, which is again by BIRAC. Then you can check Sitare. There are different kinds of fellowships available with government. So you can avail those fellowship, explore that particular segment, and then decide whether you want to do a startup or no. So that is, and government is boosting this a lot. They have launched various schemes to promote this. Now, the Startup India achievements. So we have around 2,700 plus biotech startups, 130 plus products already commercialized products and technologies. 6 ACE daughter funds are operational, and so ACE daughter funds are nothing but a larger pool of funds. Like I told you, the funds are also available at the maturity stage. So at this stage, there are a different kind of VC firms or venture capitalists. They all come together, pulling their money along with BIRAC, and they invest in a larger companies or startups. Then there are 50 bio incubators are established, and 4 bio clusters are formed. So which you can find in Delhi, Bangalore, and Pune. Then there are 4 regional centers, BRIC, BREC, and BRBC, BRTC. So these regional centers are also having facilities which you can avail and continue your product development. Then BioNest is a scheme for bioincubators. So bioincubators you can find in almost all states. So we are also one of the bioincubators supported by BIRAC, which is facilitating startup growth. Now, this is the strategic push by central government for the next 5 years. So there's 100 billion bio manufacturing facility hub, the knowledge transition cluster, technology propellers, manufacturing zones, which is also called as M zones, seed fund for early stage startups, annual turnover and limit increased for startups, then digital platform for IP protection, and mapping India and genetic landscape. Now there are various industries in Indian biotechnology industry. So one is biopharmaceuticals. You can also see examples below it. So Biocon, Serum Institute of India, Panacea Biotech Private Limited. So they normally produce vaccines, therapeutics, and diagnostics, which comes in the biopharma segment. Then bioservices and bioinformatics. So in bioservices and bioinformatics, there's clinical research, contract manufacturing, and contract research. So in clinical research, these vaccines and drugs are normally tested at every stages, and these bioservices company, they provide that entire end-to-end guidelines and support for doing the clinical research. Contract manufacturing is a third-party manufacturing. So if you're a startup or if you're a company, you want some other company to manufacture for you and you don't want to take that load, then there's contract manufacturing. And then contract research is when company is getting a research project from different company, and then they transfer the entire technology and research developed to that particular company. Then there is bioagriculture. So bioagriculture is nothing but developing agricultural products using different techniques. So there's transgenic crops, which is modified crops using gene tools. Then there's biopesticides, biofertilizers, plant tissue cultures. So those all comes under bioagriculture. Then there is bioindustrial, which is enzymes and biofuels. And in enzymes, we already know there are a lot of applications in diagnostics, textile, food, pharma, leather, paper. And good examples are Novozymes, which is already very famous, then advanced enzymes technologies. So these are the industries which falls under biotech or bioentrepreneurship. Now, so what is the difference between the normal journey process and the bio journey process? So in the normal entrepreneurship journey, you generate an idea, then you do a lot of screening of ideas, like which idea is much more feasible. Then you develop the concept or proof of concept, you test, then product development happens. Then when your product is developed, you do market business analysis, and then you commercialization. But when it comes to bioentrepreneurship, you first identify the need. You don't directly jump onto the idea generation, but you will go and identify what exactly is needed in the ecosystem. Then you look for possible solutions and develop the technology. Then once the solutions are shortlisted, technologies are shortlisted, then you develop the prototype. You validate the prototype, check the efficacy, and the assessment of outcome is done. So once you have all the study ready, then you take a risk of going into the clinical trials. And once it passes clinical trials, then you go to the commercialization. So there are a lot of steps involved in the bioentrepreneurship, and very crucial step is the clinical trial. So once that is clear, the commercialization becomes comparatively easier. So this is how this biotech and life science business startup process looks like. So you have an idea, then you you create your team. In the team, there's founders, there's core team, there's advisors. So when we say founders, they're the people who are ambitious about doing the business or starting the company. They become the founders or stakeholders of the company. Then there's core team. So after founders, there's core employees, which are very crucial to the company for developing the technology and also business development. So those are the core team which are involved in the team creation. Then advisors. So you need advisors to guide you at every different path to help you understand what exactly you need right now and what you should not look for. Right? That is very important at very early stage. Then you design your business model, you validate your business model, whether it is going to work. In business model, there are 2 to 3 aspects. One is you directly do business to business, you also do business to customers, and you also do business to customers, right? So in business to business, you deal directly with big companies who will be taking your product in the bulk. And then in B2C, you sell your product to individual customers through various channels. And then in B2B2C, your end users are the retail customers. So you sell your product, your end users are the normal consumers, but you can't directly sell it to them because that requires a huge effort. So you take one mediators in between, and that mediators will sell your product on your behalf. So that is B2B2C. So once that is done, you validated your business model, then you go for IP. You file an IP so that your technology is protected when it is in the market. And then once the IP is done, the fundraising process starts. So now you need fund to scale and manufacture. So you look for different kind of funds. These funds could be government funds, angel investors, in VC funds, depending on the need and the quantum of the fund is needed. Then there's manufacturing and distribution of your product. That is the last process that you look at. And then parallelly, there's drug development happens, animal testing happens. Then you go to clinical trials, phase 1, 2, 3. You check for regulatory approvals, what all approvals and certifications you need. And then you do those 2 activities parallelly. Correct. An end goal of any company is to sell your product and become that big so you can launch an IPO, and then your company becomes public, and public becomes stakeholders in your company. Yeah. So now this is one stage which you can look at. So you normally do project evaluation, you develop your business concept, make a plan, you build your team. And then you need finance here. So here you normally take finance. Either founders invest in the company, or there are angel investors who are there to help you or invest into your company and believe in your idea. Then you take that initial funds. Then you do foundation product development. Then you build your organization, develop your business. Then again, you need fund to expand, right? So that time angel investors may or may not invest, because the quantum you require at that stage is little higher than what you needed in the very initial stage. Then again, for business angels and venture capitalists, once you grow further and you raise another round, they take exit. So when you say exit, it means they sell your shares, and then they earn profit against that shares. So this is again a detailed landscape for life sciences startups. So again, we discussed, team is very important. So you have your founders, advisors, and experienced executives. Then you develop your business model. Then you will ask a lot of questions at that stage. Parallelly, you also try to reduce the risk associated with your technology. So you do a lot of experiment, generate data, and you file a patent. So your technology is protected. You form a company, then you do fundraising, which could be crowdfunding, angel investment, VC investment, depending on the quantums. Then one way is to license your company to different companies and earn royalty on it or profits on it, depending on what agreement you signed with that company. Parallelly, you also do risk reduction. So not entire risk is on you. So you develop partnerships for that. So you will have a partner who can do clinical trial for you. You have partners who can do manufacturing for you. You have partners who can generate data and present and collect it. Then you parallelly have your operations team who's taking care of entire product development in backend. And then again, the end goal is IPO or acquisition. So acquisition is your company is acquired or taken up by some other big giant, and you become a part of that giant company. So now what happens at university level when you want to do a startup? So you go through different trainings and training support. So you will be trained on different technologies, business part, creating pitch deck. Then you get a project support from the university. So they will give you some funds to develop your project. Then you also take services as in you give your samples outside to get it tested. Then the second stage comes at pre-incubation. So in the pre-incubation, your ideas are very early, right? You are still exploring various solutions to tackle that particular problem. And then that's where the mentoring comes in. Then comes the incubation part, where incubation helps with everything, because incubation has that ecosystem built already for you. So you don't have to go out and look for different kind of services. So in incubation, you will get project mentoring. You also get help and support in developing your product, and then also the commercialization of the product. Then there's acceleration. So acceleration is once you have the product ready, or you have minimum viable product which is ready for market launch. Then acceleration happens, where your entire growth is doubled in terms of commercialization, product certification, or funding if you need, infrastructure if you need. If you need to go through various channels, then the acceleration comes in picture. They also give you an access to their network, so that you can expand for very quickly. Now, this is the recent headline. So there are new startups which are generated in around 1,128 startups were formed in 2021. And Maharashtra is also emerging on top, and Telangana and Karnataka are already there. So these Karnataka and Telangana is already a hub for biotech startups. Telangana also have this Genome Valley there, which is a cluster of all big biotech companies. It's like Silicon Valley for startups, for biotech startups, right? So Maharashtra is also coming on top. Now, these are some of the examples of biotech companies who were once a startup, and now they are doing really well and already established company in India. So this Forest Health, Biocon, C6 Energy, Bharat Biotech. Now, how this riddle is helping this ecosystem is we are supporting innovators and we are accelerating their growth. So we are supported by DST, DBT, BIRAC, and also the state government, which is Maharashtra State Innovation Society. This is the impact that we have created so far. So we were actually operational since 2010, but in 2016 we officially registered our company, Section 8 company, which is a not-for-profit company to support innovations and startups. So we have supported more than 160 startups, created full-time jobs, around 600 plus. The total valuation of our startup is around 1,226 crore. Then total revenue generated by startups are 290 crore. And we have already given more than 115 internships every year. We were featured and have received awards more than 150. So we are not only local, but we are also global. So we have people coming in from 41 countries so far, who are part of our programs, events, and also our ecosystem. We have trained more than 17,000 students. We have helped researchers, published 17 research papers. We also do various events called Maker Mela and Darwin. So in Maker Mela, we facilitate makers from different fields, and we give them platform to showcase their product on the on Maker Mela platform. This is how we are fostering active participation within communities. So we host a lot of startup events, which is, we did National Startup Day on 16th of Jan this year. We conduct a lot of workshops for universities and faculties to train them in entrepreneurship, and also help them develop that skill and cultivate this particular mindset into a student. Then we also do training programs for faculties, which is supported by MSFDA. Then we were part of Maker Fest, which happened in Kerala. We are also mentoring EUA Center of Career College, which is in Bhopal. And we have done a lot of partnerships to boost the ecosystem. So we work in a more collaborative way than an individual. These are some of our success stories. So two of our startups have got acquired by Miko and eBiko. We have recently taken an exit from one healthcare company called Rimo. So the Rimo is into rehabilitation part. So they gamify the entire exercise version. So when you go to physiotherapy, you normally do exercises after coming from a deep injury, right? So that is very painful for patients. So this particular company has gamified this entire version. So they will be the patient will be seeing a game on the screen, and they will be playing the game on the screen. But in the back end, they're actually doing a lot of exercises, hand movements, leg movements, their elbow movements, and the entire data is captured in the back end for physiotherapy to recommend the next exercises. So these are the government funds and schemes which is boosting Indian startup ecosystem. So that is this is Nidhi SSS. So we have got these schemes and funds from the government to help startups and innovators. So Nidhi Seed Support is a seed fund which is up to 1 crore per startup. Then there's Nidhi Prayas. So Nidhi Prayas is given to ideation stage startup who wants to develop their technology, and they don't have funds initially. So Nidhi Prayas is up to 10 lakhs per startup. Then there's Nidhi EIR. So Nidhi EIR is Nidhi Entrepreneur in Residence. So this is normally given to startup on a stipend basis. So they receive a stipend every month to work on the project. Then BIRAC has a lot of funds, BIRAC Seed Fund, BIRAC Big Grant. Then there is LEAP Fund. So BIRAC Seed Fund is for startup who are at a little early stage, and they want to launch their product in the market. So this is the seed fund is up to 30 lakhs. LEAP fund is up to 1 crore. Big grant is more like a grant which is given to startup to develop their initial technology, which is up to 50 lakh. Then there is Startup India Seed Fund Scheme, which is again given to startup at early stage. So this is 20 lakhs as a grant and 50 lakhs as convertible debt. So this is it from my end. If you have any questions or queries, you can directly reach out to me, or I can take questions now if there are any.
Thank you, ma'am, for the informative talk. So dear participant, if you have any query, you can unmute yourself and ask to the ma'am. I am also very thankful to Ashish Rai to connect me with you. Actually, he is my junior, and they has doing very tremendous job in Bansal Institute of Technology. They are promoting biotechnology. They have also got several awards and several type of funding from the various government agency, the biotechnology area, and EUA fellow, that is SSYU. So they are also in they also got the EUA fellowship, and we are also established BTech Biotechnology from this year at Calcutta University. We do not have previously the BTech Biotechnology program in our university. So we have run this program from this year. And ma'am, we have also a functional incubator registered as a Section 8 company, and more than 100 startup has been incubated at Galgotia University. 16 among the 100, approximately 16 is earning the money of more than 50 lakh rupees and incorporated in MCA. So ma'am, we need your support to...
Definitely. If you need anything, do let me know. We'll be happy to help. So
dear participant, please unmute yourself and ask if you have any query. This is the session for the question answer. Any query? Vineet, are you here?
Yes, sir.
Yes. So got the opportunity here.
Yes. So, ma'am, I am also working on a startup which is in ideation stage. So my aim is to provide practical education, a platform which will aggregate research providers with research seekers. So like you were saying that in your lab we can perform our research or do like drug discovery or these kind of things. So how can we approach your avail these services?
So you can, if you want to access the facility which is available with us, you can check our website. I'll just drop the website link here if possible. So you can check our website. You will find list of equipment and facility that we have. And if you need that, you can directly email it to us regarding your need and requirements. And then we will let you know further process. It's very easy. You just have to book the machine and you can come and perform the experiment.
Where your facility is located?
Yes, you can continue.
In Maharashtra?
Sorry?
So where are these labs of your located?
This is in Maharashtra, Mumbai city. If you are not in Mumbai or Maharashtra, you can let me know which state you are from. So there we have our collaborators in every state. Bionest incubators are everywhere in India. So I can connect you with that particular incubation center where you can go and do your product development.
Okay, ma'am. Thank you.
Yeah.
By the way, I am from Delhi NCR region. So I will if there are any incubators in this region. So I would love to know that.
There is FIT in Delhi. It's an IIT Delhi campus. So they are also open for having startup at early stage. So you can check their website. And there are
FIIT.
At IIT Delhi. Then there is IndiGram if your startup or research falls under agriculture. So the IndiGram is also there in Delhi. And then there are various new incubation centers have opened in Delhi. So, you can also check Bioriidl website, and there you can find the incubation center as per the state. So you will find a list of all those incubators there.
Okay. Yeah. Thank you.
May I?
Yes, please.
Yeah. So thanks for that informative session. Actually, I just want to know that as you're mentioning that it's a DIY lab, do it yourself. So for that, I think we need to have prior knowledge of handling the machines, or you are just providing that as well, help as well.
So what we do is we first try to understand whether that individual has that expertise of handling equipment or no. If no, then we give them the training. We organize training for them. Then we make them aware about how to use that technology, how to use that particular machine, everything. We do all the training for all equipments, and then we ask them to start your project. And if there is any difficulty in between, also we have technical mentors who can help them overcome that difficulty.
Okay, so for that training, so do we need to pay extra or what?
So trainings, normally here what we do is we take this membership model. So if you are here and want to try your project and idea, so you can take membership. So training and access to all the lab and facilities included in that. And membership is we have kept it very low to maintain the lab, so everyone can get access to it.
Okay, because I am hearing for this Bioriidl for the first time, so I just want to know that is this the only revenue generation model that you have, that you are providing the facilities, or what are the other aspects that you are catering to?
So we are, Bioriidl is facilitating all kind of bio startups, including food and healthcare also, and even innovation. So we also have, we are running research groups to help these people understand what exactly they want to do. So we have 5 research groups themed. So people who just want to do research and doesn't want to get into startup, but they still want to do research and commercialize it. So that research groups are there for them. Apart from research groups, the membership that I just talked about, so people take that membership to understand whether they want to do a startup or no, right? So they just want to first try out their ideas, then see whether it will be a good fit for the market or no, and then what risks are involved in doing that, whether it is feasible or no. So there are various things that is normally answered during the membership duration. So they complete that product development, and then whether if they don't want to continue with the startup part, then they normally apply for licensing that particular product which they have developed, and then we connect them to different companies accordingly. And if they want to take it ahead as a startup, then we enroll them into pre-incubation part if they are very early. And if they are ready with their MVP or minimum viable product, then we involve them into the incubation program, where they will be exposed to various sessions like business development, a business plan, financials, legal, everything. So they will by the end of the program, they will know how it works, what they have to do, and how they want to take it ahead. And we also have some funds associated with our incubation program. So we also give them some internal funds, or we also have government funds. So we ask them to apply for those government grants and funds. And then if they get the fund, they take it ahead and develop their product.
Thank you so much. And I'm looking forward to contact you for that. So thank you so much.
Yeah.
Ma'am, actually, we are now EUA will also open in this year. So in previous year, our 3 teams get shortlisted and presented their idea in Punjab University. Okay. So they didn't get a selection, but actually I need your advice to how to what should be focused area should be chosen by the PG and UG student for the EUA fellowship as well as for the big grant. Because in the EUA, I think the faculty member has forced the student to take a vast or complex type of project. But I think the all recipient of EUA got selection due to the very simplicity of the project that can be turned into a product. So I need your advice. I need your comment on this.
So normally what happens is EUA is given to students to develop that entrepreneurial mindset right from the start when they are in their career building level, right? So EUA doesn't mean that you have to have very complex problem to be solved, but it is more of you are solving a problem which is need of an hour, even if it is already there, but you create that niche for you in the problem. So maybe everyone is producing vaccine, right? All companies already know how to produce vaccine. but you are also producing the same vaccine, but you are maybe easing the way of giving that vaccine, which could be your differentiation from them. So you just have to find that particular differentiation. You can work on the similar problem, you can develop the similar solution, but there has to be one entry barrier, or one thing that only you are able to do and others cannot, or they might take time to develop that once they see your product. So that one niche has to be there. Otherwise, it's not compulsory that you have to work on the complex problem. You can take as simple project, but develop that particular project into something which is not easily copied by others. So that one thing you have to keep in mind. Rest, I think if you have a very simple solution, but also have very one IP thing that you can protect that particular solution from others, that could go a long way compared to the complex problems, because the complex problems takes a lot of time for validation.
Thank you. Any other query from the participant? No query. So it's time to felicitate our speaker. So ma'am, on behalf of Galgotia University and organizing committee, kindly accept a memento of honor for the gratitude. So for delivering such a wonderful, interactive and informative session. We are highly obliged to you, ma'am. So thank you so much, ma'am.
Thank you so much. I'm glad I could contribute something here. So thank you. If you have any queries, you can reach out to me at my email ID. So you can take my contact details from Galgotia's University. Yeah.
Thank you. Thank you.
Thank you.
Okay, we are moving ahead. Our next speaker has joined with us. Debnath sir, are you here? Good morning, sir. Debnath sir.
Can you able to hear? Am I audible?
Yes, sir. Yes, sir.
Hello.
Yes.
Okay. Yes. I am not sure in which name I have joined.
Okay. I will rename your name.
Okay.
Yes, sir. You can on your video also, sir.
Okay, sure.
And I have made you the co-host. You can share the screen. First, let me introduce. Yes.
Yeah. Please go ahead.
Yes. So, dear participant, our next speaker is Dr.Surajit Debnath. He is the head Department of Medical Laboratory Technology, Women's Polytechnic, AICTE, Tripura, with more than 13 years of experience in biomedical research and high-level teaching. Dr. Debnath participated in various academic work in Institute of Germany, Denmark, and Hong Kong, besides in premier India labs such as AIMS, IIT Kharagpur, IIT Guwahati, NVPGR. Dr. Debnath is the recipient of Young Scientist Award from different agencies, that is DBT, DST, CSIR, recipient of Tata Trust Grant of CME course in Royal College of Pathologists, London, and reviewer of several Scopus SCI Index Journal that is published by Elsevier Springer and reputed publication house, along with the reputed and regular academic activity. He is also engaged with the several biotechnology companies in northern and western India as a consultant. he have conducted various workshops and talks, acted as a session chair in international conference sponsored by the national and international academic agency. He has delivered various invited talks in the national and international academic gatherings. Currently, he is guiding a group of international students for their internship from the Russia, Turkey, and West African country. Dr. Debnath filed 2 patents in recent year in India on biotechnology. So we are very grateful to have you, sir, as an invited speaker, and we are inviting you to deliver your talk, sir. Over to you, sir.
Yes, thank you. Yes, thank you, Gaurav sir, for this introduction. I'm again thankful to the organizers of this whole event, including DST, Department of Science and Technology, Government of India, for sponsoring this such a wonderful interdisciplinary session. By saying so, I would like to, sir, can I able to share the screen now?
Yes, you can share.
Okay. So what exactly we will do is I'll share the document and just give me a quick this thing, whether things are visible or not. Just hold on. Just let me know whether the things are optimal in visually optimum as well as audibly correct. Can you able to see, sir?
Sir, currently your screen has shared, but we are not able to see your
Okay.
So you are presenting via mobile.
I'll make it sure that it is not compromise the quality things. Just give me a quick feedback once automation. Okay. So yeah.
Sir, you can also tilt the mobile and get it to the full screen.
Absolutely. Gaurav sir, just give me a quick feedback whether the things are optimally visible, sir.
Yes, sir, they are visible.
It is visible, right? Okay. So good afternoon to one and all. So what we'll do is I'll request some of our from some of our participants to act as volunteers. Okay, once we go ahead with this. So anybody who would like to volunteer during this session, you can put it your name in the chat box with our coordinator, sir. To become this thing, you need to join through your your desktop or computer or laptop even. Okay, just let us know. So what happens, we are talking about this thing that how to automate your enterprise goals by using artificial intelligence. So in an ideal situation, the whole discussion could be done in 2 parts. In the first part, what we can do is we can find out what are the different tools we can use for a particular kind of bioentrepreneurship goal that we may have. So give you one more quick this thing of ours, that in my lab we basically are doing work which is mainly on in vitro diagnostic tests. Okay, so we are bigly into it. We have developed 2-3 protocols and products as well, and we are trying to market those these things, but we have not developed full scale into AI, but we are in the process of it. So today's discussion would be more on how we can streamline the whole process and why exactly we should do it. Like in the previous session, I am sure that the speakers must have discussed those things. But I would also like to focus on how the diagnostic industry in India is working and what are the long-term goals. So Gaurav sir, once again, would you please give me this thing, quick feedback that whether the things are optimally visible and audible, sir? Or any one of you can just let me know. I cannot see the chat box. You can unmute yourself and you can tell me. If anything, this thing is not up to the mark, just let me know. You do a short this thing, because my institute is subscribed to a different app, but this particular program is going on in a different app. Participants, things are okay? You can see?
Yes, sir.
And
you are on
audible.
the first slide.
Okay, fine.
I will change the
So what
slide,
we'll
sir.
do? Yeah,
Yes, slide is changing.
So what has happened? Okay. So what has happened? This discussion, you could consider that this is the first part of this whole exercise that we intended to do with you all. This is the first part scenario. What we will do? We will do an overview of the pharmaceutical industry. We'll focus on this diagnostic scenario, that how the market is evolving and what are the technologies we should incorporate. Then subsequently we will move on to tips and bits about what artificial intelligence means and from where it has been came. So basically we will be talking about the infrastructure that could be built on AI. So recently we have come across the McKinsey company data, which is a global R&D market research organization, and very authentic data they produce. So what has happened is you can see this graph which indicates that the amount of capital that has been raised by the pharma company. Okay, so you can see from Q1 of 2019 up to the Q1 of 2021, there has been steep rise in the funding in these pharma companies. Okay, so you can see just after the 2020, it has been practically doubled. The amount of financial basket these companies are having today, available money that they can deploy in R&D and all other things, is huge today. Most of the pharma companies, including India. So this particular scenario includes US markets, but a substantial amount of Indian market is also represented in this snippet. So what we are interested in, so these amount of money which is already there with the pharma companies, where they are going to deploy it, they are essentially going to deploy it into R&D. Okay, so what happens in pharma and this healthcare sector is the sector which is very capital intensive, and there is also a good return. I think all of you have heard of blockbuster drugs. These blockbuster drugs are drugs which is patented by a company, and it is exclusively selling right for a particular company for approximately 5-6 years. So in 5-6 years, what happens now? Most of the business is being made. Then what they do is they license their protocol to some other generic companies, and subsequently they get benefit from there as well. So it's a huge market. So pharmaceutical market is also fragmented into multiple things. As the earlier speaker said about vaccine design, vaccine development, this is one market, biopharmaceuticals. One more market is chemotherapeutics, that is small molecules, and then what else? These antibiotics and so on. The other market is diagnostics. So we are focusing on the third part. Okay, so here is some other interesting scenarios as well in the same report that has been published by McKinsey. So what it says is that the growth input drivers of these things are very high these days. So what has happened? Initially, there was an expectation that there will be 7.3% real GDP growth. It has been increased into 7.9. Means what? Means people are having discretionary income, please understand, to deploy. Are you getting? So what these people will do with the discretionary income? They will go for a thing called preventive healthcare. Okay, preventive healthcare is what? To do some diagnostic tests in advance, so that you can predict whether you are prone to some kind of disease. It includes heart disease, lifestyle diseases, and multiple different things. So we are trying to make you give you this thing, that why we should look into the pharma sector as well as this all the peripheral sectors that is there in the pharma in a big way. because investments are there, the money is there, and there is also a what do you call significant opportunity in R&D as well. Okay. So these are again a split of the market by type of opportunity, as you can see. So there are, in aggressive case, what would happen is there will be a 50% increase year on year in this particular what we call this particular pharma market, which can be explored by people who has been trained in this particular domain. So if we see this snippet, which indicates that what are the different divisions of the pharma market, as you can see, there are oncology, there are immuno-oncology, there are vaccine design, there are biopharmaceutical. One part is diagnostics, which is big in demand. So we'll come to those things. So one more interesting snippet is this. Here what it says is the amount of intellectual property that has been built in last couple of years. Okay, from 1992 to 2016. This is a limited this thing, but this is going on. So what this snippet tried to tell you is that there is also a boom taking which is called this artificial intelligence, machine learning, and all such domains. So there are two pictures. One, there is a huge demand in diagnostic industry, in all sorts of demand, and people are having their discretionary income to be invested in this particular domain. Also, the pharma companies are also sitting on a huge cash which can be deployed in R&D. So this is one scenario. Another scenario is there is a rapid development in this particular sector, which is called this computational technology. Okay, microprocessor based technologies, data science, then artificial machine learning, and then finally the artificial intelligence, natural language processing tools and all. Okay, we'll come to those points. So in our lab, in a small way, what we are trying to do is we are trying to amalgamate between these two sectors. Okay, to come with some amount of what we call novel intelligence, or shall I say intellectual property that we can market. Okay. So this is one more interesting scenario. Now this data is produced by Deloitte. Deloitte is again a global R&D firm, which shows that there is an increased demand of those technologies which are digital. So what they're trying to say in this snippet, if you go in detail, you will see that diagnostics coupled with digital is big in demand. Okay, so there are multiple verticals to it. Okay, I'll give you a small example. Let us talk about this IoT devices. Okay, or smart connected devices. You are running in a particular park, and you can also see how much of calorie you have burned. You can also see what is the cardio output that particular park. So we are not into that, but we are somewhere similar. So I'll just give you an idea. Now, this is the diagnostic patient care scenario where multiple different things are there. One part of that is in vitro diagnostics. Okay, so we'll also tell you that how in vitro diagnostics has changed over the years. Now, this particular snippet will show you how the integration between digital and wet lab things are taking place every day. Okay, so I will not dig detail into it. Now, this is again one more snippet which indicates that market creation during the next decade will involve collaborating with broader set of partners to enhance diagnostic rates, compliance, and thereby changing the very nature of the what we call patient funnel. Okay, so what they are trying to say? They are trying to say this is not our data, this is third-party data, which says that if somebody can get into this particular domain where digital is combined with the conventional in vitro diagnostics, then it could be in huge demand. Okay, so this is again a similar snippet which gives you this idea that how exactly people can do it. There are various verticals on which we can work on this particular project. Okay, Now, this particular snippet gives you an idea that how artificial intelligence is developing day and night. Okay, so it is basically on top of the pyramid. Okay, now this particular figure is in verticals, but if you can see it from top to bottom in an upside down scenario, then you can see that artificial intelligence or AI is present on the top of the pyramid. Now, what is the base? Now, base is something called data science. Now, data science is nothing but the ability to predict on the basis of some training set. Okay, very simple idea to understand. So, what data science says? Data science says that by deploying statistical tools, we can have an ability to predict the future outcome if we are having a very good training set, which is classically in statistical language is called population size. If your population size is good, what you can do is you can predict with very high degree of probability that what could be the outcome of a particular event. Now, data science, when integrating multiple types of statistical tools and analytical tools, it gives into or it enters into another domain, which is called machine learning. Now, machine learning is not very different from human learning. So, how human learns? Human learns from its experiences. Okay, I give you an example. If one of your teacher teaches you that this is how you should program your computer for a particular outcome, then what you will do? You will do the same, and subsequently you may able to generate a different kind of analysis. Or okay, so in this case, what happens? Since your background has been built in a supervised way, you can tell it as a supervised learning process. So machine learning is also this thing divided into some very different types of learning process or learning algorithms. One is called supervised learning, where there is something called supervision under which you develop your learning process. One more process is called unsupervised learning, where nobody teaches you, you learn all by yourself. That means what? You generate a data, and on the basis of the data, you design your algorithm. Now, one more type of this thing, learning is reinforced learning. In reinforced learning, what happens? Every time you learn new things, you increase your what you call as the complexity of the algorithm, and eventually you end up with something which is called deep learning, where you can connect multiple faraway dots to build a bigger picture. Okay. Now, in deep learning, there is one more dimension into it, which is called artificial neural network. Now, artificial neural network is further divided into multiple different types of networks. Okay. So, what does it mean? It means that artificial intelligence is based on all the fundamentals of data science, machine learning, and all sorts of computational intelligence into a very higher possibility. In the hierarchy, the topmost position is for artificial intelligence. Now, it is our job how to deploy artificial intelligence for diagnostic industry, and what would be the outcome of it. Okay, the major outcome, as you can all understand, is reduction of cost. Okay, so I give you one small example. What a coordinator, sir, just let me know whether everything is audible and visible. So what happens when we deploy these tools? What happens? You get a drastic reduction in your cost. Why? I give you a small example. We will do a little hands-on if time permits. If some of you would forward as a volunteer to whom I'll share some files, they should just only have to share their screen and do the things as I say. They're very small tools and very easy to handle. Anyone can do it. Okay. So what happens? Just let me know. You can hear me correct, and you can see the slides in the way I want. So what happens?
Yes, sir. Yes, sir.
Okay. Okay. So what happens? Human learning is very costly. I tell you. Consider that we are having a collaboration with a tertiary hospital. In the tertiary hospital, there is a department called cytogenetics. In that cytogenetics, there is a person who has done his MBBS for 5 years, then did an MD, then subsequently moved for a certificate course in UK, and then came back, and then 15 years of practice, and now he is the head of a particular department. Now you see how much of investment was there for this person. Now what is the function of that person? Please understand. He every day sees biopsy slides in order to identify in which stage the cancer is. Please understand. By looking at a biopsy slide, he can tell me whether polyploid is there, whether metastasis is there, and small things. Okay. So this is the expertise he has developed over the years. Okay. Now, what do you think? What is the biopsy slide cost? It will cost you approximately 6 to 8,000 rupees for every slide to be investigated under him. Why? Because he has invested a lot of money, time, intelligence, and all such things to develop an expertise which is indispensable. Understood? Means you cannot do away with him. आप उसको निकालोगे तो आपका hospital ही बंद हो जाएगा. Understood? So he is the moat. As the previous speaker is talking, I do not know whether you have marked her point or not. She was essentially talking about something called moat. Moat is which is an USP of your company, an entry barrier for this particular hospital with which we are collaborated with. The moat is this person, this professor, who is so good in identifying biopsy slides that nobody can do away with him. So he is the moat of the hospital. He is the best one to identify in Northeast India. I belong to North. In Northeast India to identify biopsy slides. If he says, okay, this is the diagnosis, AIMS also says same. New Delhi AIMS. So you're getting, so this is how this thing has been developed. So what would happen if you want to replace it? So what you need? You need to have a huge amount of database upon which your algorithm will get trained. And eventually what would happen, you know, by a single click, you can differentiate between a primary carcinoma and a tertiary level carcinoma. Okay. Okay. Now, how we'll do it? Now, the whole data science picture is divided into 3 things. These are the 3 fundamental things. You can take a snippet out of it. That these are the 3 things which should be there. One is classification algorithms. Second, regression algorithms. And third, a clustering type of algorithm. So all things are divided into these 3 things. So you can ask me, sir, what is this? So we can do a small hands-on if you require, if you want to understand what exactly regression means. Many of you are already aware of it. If anybody wants to do the hands-on, you can just tell me. I'm sharing this thing, what do you call an Excel file with you all in the chat box itself. In Zoom, just give me a second. And if at all, you can play with this also. It's a small this thing. Just let me unshare this. And I do not know how I'll able to do it in a nice way. Just hold on. Give me a little time so that I can share by you. Just have it. If you think that okay, you can do it this hands-on, because sorry.
I think due to the bad network connection, sir has terminated from the meeting. So I am contacted to rejoin.
Okay, so I'm just sharing a file with you. File sharing is just for the where I can find the file sharing. Okay, with participants. Yeah, I'll just Gaurav sir, we cannot share files here, right?
No, no, you can share.
Or is this okay? In
The chat
which
box you
option?
can attach
Okay,
some file
it's nothing.
if
It's
you want.
okay. Yes, yes. I'm trying how to. Most then participants. Share. Gaurav sir, please direct me after going to the chat box.
Yes.
I'm finding all the list of all
Yes.
people.
In the chat box, there is a small icon. You can see. One will be the format. Second will be the screenshot or file. So just click on the file to share the screen. Share the file in the chat box. Or you just send to me. I will share
Okay, fine.
on your behalf.
Yes. Yes, yes. Found, sir. Thank you. It's a small Excel sheet just to give our participants an idea. I hope it has been, yeah. Yeah. To give our participants an idea that how data science works. The file has been received, sir, Gaurav sir. One Excel file.
Yes, yes.
Have you found?
One, yes.
Okay, okay. Okay.
One
Is there
Excel
any
sheet, no?
participants who can?
Yes,
Yes,
Excel sheet has been.
who can? Okay. If any participant want to coordinate during this process, it will be great, because I do not know whether Gaurav sir will permit it or not. It is just to make our participants to see that how the basics are so easy to understand, but to design the whole architecture, it becomes a limiting factor for those who have been trained in biomedical sciences, and therefore we need computer science people for collaboration. The file is not opening.
Yes, it is open, sir. Please.
Oh, I think it is opened. Dr. Sweta, please see. I think it will be open. Gaurav sir, is this possible that we can do this by sharing your screen?
Yes, yes.
Okay. So, okay. If Gaurav sir, okay. Thanks. Okay, so all of you, as you can see, it's a very small data. What is the data? There are two X and Y, what you call axis. Okay, in one axis, there is a thing called LH. So luteinizing hormone is a biomarker, if I am audible to you, which indicates the development of breast cancer. Okay. Now this is there is no intervention needed. You just need to isolate the serum, then put it into an ELISA reader, and it will find it out what is the concentration of luteinizing hormone in your blood. If it is within the normal level, absolutely fine. If it is not, it indicates that there is metastasis in the breast tissue. Okay. So one more interventional process is there, which is called biopsy. In biopsy, what happens? In biopsy, somebody will put a needle, which is called fine needle aspiration cytology, FNAC. Through a needle, some fluid or tissue will be collected from internal part of the body, then put it into a slide, then stained by a particular thing, then again observed under a microscope and a high power microscope, it could be fluorescent also, to identify the bending patterns as well as the number of chromosomes that are there. So if the numbers of chromosomes are more, it indicates polyploidy, which is again an indicator of cancer. So here, this particular snippet, what it is trying to do, it is simply trying to find out a way so that only by blood test you can confirm that the polyploidy will be this much. So what is the idea? The idea is to do away with FNAC because it's a trauma for the patient. Okay. In order to prevent the trauma, what you are trying to do, I'm just giving you a small example. What you are trying to do, you are trying to generate a correlation between the serum biomarker to a cytological biomarker. So what you can do is you can share both the two columns, okay, and then make it into an XY curve, okay. So what you can do, you can share, you can Gaurav sir.
Yes, sir.
Both the columns can be selected, and then we will go to insert.
Sir, we have to select the both column.
Both columns. Yes, sir.
Okay.
Then we will go to insert.
Insert.
Then we will go to insert.
Okay.
Sir, insert, and then we will go to what is that? What is that? Okay. No, that is a different one. Okay. Here the top one, sir. Top one, sir. Top one of this XY curve, sir.
This one.
In the chart section, sir. In the chart section, we shall go. In the chart.
Okay.
A little bit this right side. Chart.
Okay.
Right. Oh, top one, sir. Top one. No. Just above this. Just above this, sir.
This one.
Just above this. No. No, right side. Sir. That side. Right side.
Pivot chart, sir.
Right side. Yeah, this one, sir. This one. This one.
Okay.
No. That one, sir. That one. XY. Next to that, sir. Next to pivot chart. Sir. Top. उसका ऊपर में, उसका ऊपर वाला सर. यही वाला.
Bubble chart.
हाँ. Please click on this, sir. The first one. Yes. Thank you, sir. Thank you, Gaurav. Thanks a lot, Gaurav, sir. Now, sir, we'll add the trend line. We'll click on the dots, and all the dots will be selected. We'll click on the dots. Any one of the dots. Okay. All the dots will be selected. Now, sir, left click. Add a trend line.
Left.
Sir. Oh, sorry. Right click.
Right.
Sir, after selecting the dots.
Yes.
Then we'll just now, sir. Once again, sir, select the dots. All the dots. Once again, sir. All the dots, sir. No, sir. Previously, by selecting one only, all the dots will be fine, sir. Now, sir, left click, sir. Simply. Then it will come add trend line.
No. So what we have to do?
Trend line will come. We have to now left click, sir. Inside this thing only. So, sir, one option will come out. Sir. Add trend line. Third option.
Okay.
Last but this one. Yes, sir. Thank you, sir. Okay, the trend line has been set. Okay. Now this trend line is a straight line. So y is equal to mx plus c. It means what? It indicates that these two parameters, which is directly proportional relationship. If the luteinizing hormone international unit per ml is increasing, number of polyploids will also be increasing. Okay, so it means that it's a straightforward relationship. We can even insert this thing also. What you call is that equation also. That equation, if you have a unknown concentration of LH, for example, one patient is having an unknown concentration of LH, which is approximately, let us say, 250. Okay. Now in 250, if somebody's LH concentration is 250, what will be the number of polyploids in the FNSC slide? You can directly deduce from the straight line. Are you getting my point? Now this is called trend line predictions. Particular thing is just for a your understanding. Thank you, Gaurav sir. Thanks a lot. I hope our participants
Should
have
I
understood.
stop the sharing?
So I'll go back and now I'll put my this thing. Yes,
Should
sir.
I
Yes,
stop?
yes.
Okay.
Thanks a lot, Gaurav sir.
Okay.
Yes, sir. Yes, sir. Yes. So now what we'll do is, now we'll again start our discussion. Okay. Now we have okay. Now this is the fundamental of all your data science problems. So if you are good enough with this particular thing, you can also understand how the prediction or how the machine learning thing happens. Understood. So what is our job now? Our job is to this thing, the dots, okay, to connect the dots. So my lab is analyzed in vitro diagnostics. Okay? We do in vitro diagnostics on regular basis, all our students, every day and every night, of all sorts of this thing you can think of, okay, including RT-PCR and all. So what happens? We generate a lot of data. So what is the use of the data if your this thing is not yet customized? So we are trying to build some customized solutions. Let us see those. We may not be able to complete the whole thing today, but okay. Now this is the basis of this. Now in supervised algorithms, there are some special kind of algorithm. I'll talk about only one because we'll do one more hands-on, which is entirely based on this particular thing. Okay, these are the supervised algorithms which are very common and very easy to deploy. We'll see today also. So these are the main supervised algorithms, which is called bias algorithm or a probabilistic classifier method. One is called decision tree, another is called nearest neighbor joining, and another is called support vector machine. Okay, so these are different classes of supervised algorithm. Okay, now out of all the supervised algorithm, there are some data that which kind of algorithm is good for cancer prediction. Okay, so we have found out from literature search and by talking to our other experts globally that this particular tool has been given excellent result to identify what you call mainly for cancer lesions if you have a sequence protein or genome sequence. So what has happened? What is this probabilistic classifier method? PC. Now this is a supervised algorithm, machine learning technique. Now this is based on three things. What? This is used for prediction purpose. Now what it can predict? It can predict a posterior event. Posterior event means future event. Now this prediction of posterior event is based on three things. One, the experience of the prior event, those things which has already occurred. Another consideration is the likelihood. Likelihood means what is the possibility of occurrence. Then finally the evidence. So you can see from this snippet that machine learning is not different from human learning. How human learns? How you understood that if you touch a flame, your finger will burn. Okay, it may happen that you have also tested it by yourself. It may also happen that your parents might have told you. Understood? From where? From the prior experience, from the evidence and the likelihood that it will burn. If you put your finger in the flame for this many times or this many seconds, you will definitely get your skin burned. Are you getting? So this simple is the whole technique. So we'll also learn that how exactly it will predict and what are the outcome looks like. Okay, for particular breast cancer we'll be talking about. Now there are some limitations also. We are not discussing that. One more algorithm which is very easy to understand and deploy is called decision tree. Okay, we will not also go to this thing. Now in decision tree, how it works? If you see this snippet, you will understand. Very easy to understand. Consider that one person is there who have detected a lump on the breast tissue, which is done by usually self-examination or by a trained person. Then what would happen if the lump is detected? The doctor may tell you to go for a mammogram. If the lump is not detected, there is no need for a mammogram. Mammogram is nothing but a what we call ultrasound scan of the breast tissue. Okay. Subsequently, if the mammogram is positive, if a mammogram is not any significant, the doctor will say, okay, nothing to worry, you go. If the lump is detected and mammogram is also positive, then the doctor will tell you to go for the FNS or biopsy that we are trying to do away with. Please understand. Then, if the if the cyst is detected by biopsy, if the metastasis or polyploid is identified along with large luteinizing hormone concentration in the serum, the doctor will tell you to go for lumpectomy. Means what? The lump should be this thing do away with. Okay. So this is how the decision tree works. Okay. Now our job is to customize the whole thing for a particular patient. So we will not discuss this particular example. There are multiple different algorithms which are being built on this particular thing. How to do away with the interventional process, where you need to do a local anesthesia or a general anesthesia, then do the diagnosis. You are not doing any operation; you are just doing the diagnosis. Okay, these are very difficult to because the patient compliance is very low. By looking at the biopsy process, the patient will go, okay, I will not go for the any more diagnosis. Let it be however it is like. So this is not a goal, right, for medical fraternity. So here we are showing you two things. One, the conventional way of diagnosis, where, as I have already discussed, the other one is the deployment of artificial intelligence tools. The bottom one, the top one, the existing one, the bottom one, the future one. Okay. Now this is one more snippet which gives you an idea that among all the supervised and unsupervised algorithms, which are giving good results. So you can see here, naive Bayes algorithm is giving good results. Along with that, you are also getting very good results in artificial neural network. Okay, which is the basis for deep learning. Again, one more this thing, what we call a vertical for artificial intelligence. Okay. So we are also not discussing this. Now, this is a very famous example which has been published in PLOS One recently and very celebrated also. In this study, what they did is, I do not know whether you have already gone through or not. They were able to back calculate how long a patient will survive by looking only at a biopsy slide. The computer algorithm is designed in such a way, by looking at the slide only, that means the biopsy slide only, the algorithm will predict how long the patient will survive in this particular brain cancer thing. Okay, so we will not discuss much. So in order to do so, there is a thing what we call hardware infrastructure that should be built along with the software infrastructure. This is shown here. This is again one more infrastructure where machine learning and artificial intelligence tools can be deployed for biomedical research. Okay. Now these are famous individuals. Okay. So why I'm putting their pictures? We're putting their pictures because they are famous, and one more thing is common in them. The common thing is all of they are breast cancer survivors. Okay. But on top, the personality whose picture I'm showing, she is like far away between among this whole group. She is a completely different kind of person. Let me give a story about her. Angelina Jolie was very intelligent person. Other than that, she is a very famous actor also. So what has happened is her maternal line is full of breast cancer patients. Her mother died of it. Her aunt died of it. So when she found out, and when she thought that in her developing years that she could also be in danger, what she did was, she invited these Harvard University people and given a particular amount of this thing, what we call funding, to do the whole genome sequencing for this lady long back when those things were going on. Eventually, these persons, Harvard Genetics Lab, have designed an algorithm free of cost, which is available. Today we will discuss also that thing by hands-on process also, where you can predict the probability of breast cancer if you are having your genome sequence of a particular area, which is called BRCA. Okay, so this is the this thing database where you can find the BRCA reference genome. Means what? The reference sequence, unmutated, ekdam this thing correct sequence, which is like this thing of a normal person. Okay. Now this is the human genome project and human genome sequence which has been deposited in NCBI database, and then this is the BRCA gene product. Okay, the protein sequence of BRCA, unmutated, and then reference genome, reference sequence. Now, what you can do is by deploying machine learning tools, you can find out if you are having your own genome sequence for this particular thing, which you can do in any genome sequence service provider. There are many in India, in South India as well as in North India. It can cost you approximately 5,000-6,000 rupees, and you can sequence your particular gene. For example, thalassemia people, they usually go for the beta globin chain. You can even go for BRCA1 or any other thing according to your choice. So consider that you have understood your BRCA sequence, male or female. This can happen in male also. Now, then how you can deploy, how you can find it out that, okay, your mutation is a silent mutation or a non-synonymous mutation. So we'll do a little hands-on. Now, this is the whole activity that you should do before getting your whole genome sequence done. So usually this is done by wet lab service providers. They usually deploy these particular steps for genome sequencing. So the WGS sequence analyzers are usually by Illumina, Roche, or some other Bioriidl, big companies. Now the software is also deployed by them only. So you can, what you can do is by automated machine, you can have your whole genome sequence. Okay, as I have said, within a week you can get your results, and 5,000 to 6,000 rupees approximately. Okay, this is the whole process by which we are not discussing it. Now what we'll do is we'll have one more small hands-on, but before that you just see that how the diagnostic industry in India is growing. Okay, now this is the particular snippet of LPT. LPT is Dr. Lal Pathlab. This is a company which is doing excellent, and you can see the share price of this particular company. This is the share. Okay. Now this is again the share price as well as profitability of this company. Okay, Dr. Lal Pathlab. Then subsequently, this is the profit and loss statement. They are reporting year on year, quarter on quarter, rapid profit because of this preventive. As I have said, no, if you have discretionary income, what you will do? You will go for a foreign vacation or this kind of thing, isn't it? A discretionary thing. If you do not do today, that also is okay. So what you will go? You will go for a whole genome sequencing thing, or you will go for a preventive health checkup, whole body checkup, this and that. Understood? So if you have that money with you, these companies will grow. Then again, this is one more. This is okay. This is, I think, one more. Fortis Healthcare. Fortis Healthcare is also there in big in the business. This is again the Metropolis Healthcare. Metropolis Healthcare for Fortis Healthcare and this Lal Path. These are the top brands of diagnostic industry in India. So, and this is also creating a lot of jobs. Now in digital sector also, if you are good enough in these particular things, you will be having very good possibility to get involved in the growing industry. Cyber security, privacy and trust, then data analysis, data mining, cloud and data role, software development. These are the areas where you can find excellent opportunity, even biotech people. Then subsequently, IoT spendings are also increasing. As you know, in your smartwatch, you can now deploy your ECG as well as pulse rate, oxygen saturation, and all such things. So what you think? If you generate a lot of data and in some processor, it is getting processed, they can able to predict that after 10 years, what will be the outcome of your cardiac this thing, isn't it? So we are not going into that. It's another area of research and discussion. Okay, so this is again in very interesting thing. Okay, for our student, and you can also tell your students in your classroom that what has happened. Once upon a time, there was a notion that if you deploy money in R&D, your return will not come. But the scenario has changed just after the COVID. So if you deploy amount in your R&D, you get good returns. Okay, so this is now it is true. Okay, these are some work that we have done in our lab. Yeah, these are some related things where we have deployed these. These are the products we are trying to build, and we are also having collaboration with at least 2 different organizations. We have also put those things in our one innovation challenge. First round has been cleared, and the second round we are anticipating to get in. These are small things, and now we'll go for the hands-on. Okay, again Gaurav sir will come and help us in this regard. So what I'll do is I'll share 3 links in the chat box. Okay, I'll stop sharing. I'll stop sharing now. Okay, fine. Now what I'll do is I'll put it this thing. Just hold on. Chat box. Yes. Okay. Yeah. Just a second. Just simple 3 links through which you can do the hands-on, and we'll able to have a little understanding that what message you are trying to give. One second. Okay. Copy. And selecting the that. Yes. Okay. Okay. These are the 3 links sir I think has received. Yes. So what happens, as I have said, that there is an algorithm which is known as naive Bayes algorithm. Now this is known as a classifier. We have seen the fundamental 3 things are there. One, classification, one, clustering, and another one is trend line. Okay. Now this is a classifier. Okay, it will classify. Now this is based on a machine learning algorithm which is called naive Bayes algorithm. Now this naive Bayes algorithm considers 3 things. One, prior experience, then the what you call evidence which are there, and then predictions or statistics. Okay. Now what we'll do is we will deploy these tools one by one and try to figure out how this algorithm works. So what do our Gaurav sir again?
Yes.
Yes. 3 links
So,
we have shared.
Yes.
Yes.
First link should I share, sir?
Firstly. Now this is everybody knows this is a database and this is a real-time database. Means what? From your end, from your lab, if you have generated one sequence by
Yes.
proper formatting, you can also submit your link to this database. Okay. Now this is showing you the whole human genome. Okay, which has been sequenced during 1990s, and this is the unmutated reference gene. Okay. The second link, sir. Second link also you share.
Second chromosome.
No. Second link. Link, sir. Link.
Second link.
3 links I have shared, sir.
Okay.
Participants, so you can also play with it if you want. The second link. Okay. Now the second link, what we have done is we have isolated this particular thing. What? BRCA sequence. Okay. In the chromosome, no, it's like a library. We want a single page to be Xerox. So what we did, we identified the single page, the whole library, and the same thing we're doing here. What we have done, we have seen the library. Okay, let them be there. Don't disturb. We have again put one more page. We are having a gene of interest, which is the BRCA1, breast cancer antigen. Okay, fine. Okay, participants. So what has happened? We are having these 2 things. Now, subsequently, these are your data set. Now, subsequently, you need to have your tool. Okay. Now, it is loading. Okay, great. So now you are having your Homo sapiens BRCA1 partial sequence. Why? Because this is the cytoplasmic domain. We don't want the distinct domain, the transmembrane domain. We only want the cytoplasmic domain, and this is the partial sequence. Okay. Now, Gaurav sir, we will make it into FASTA because so much of unnecessary noise is there. We'll trim the data and keep it as a FASTA. Please, sir, click on the FASTA
What we have
so
to
that
do?
it is get trimmed. The whole sequence will get trimmed. Sir, we shall click on the FASTA link. In top,
FASTA.
sir, just after
Okay.
BRCA1,
This one.
you can see FAST.
Okay.
Okay, participants, as all of you already know, so what has happened? The GenPept sequence is the sequence or the data format which is giving you so much of information. What is the method you used for sequencing? Whether you have deposited it or not? On which year you have designed that sequence and all such. So we want only the sequence, not all this information. So what we did, we trimmed that noise. Noise means unnecessary information by clicking on a link, and now this is our FASTA. The FASTA means what? This is a sequence of a protein. All and each and every digit you are seeing, no, these are nothing but the single letter amino acid sequence. Okay. Now this is a protein sequence which is having 99 amino acids, small molecule, very small, 99 amino acids, and it's not BRCA1. I'll give you a little intro. It is nothing but a proto-oncogene. What it does is, if it is not mutated, it will ensure that the cell cycle is regulated in a proper way. G1, S, G2M. If at all there is a mutation, what would happen? It will not able to bind to its target properly, and eventually what would happen? The cell cycle regulation will get lost, and the breast cancer will appear. Are you getting? Very simple. Now what we'll do, we'll try to find out what a mutation will lead to. Okay, so for that we will deploy our tool. So Gaurav sir, I request you to click on the third link.
So should I copy this one?
Just let me know whether. No, not now. You just share the third link, sir.
This is the first one.
No, third one. Third one, sir. Third one.
This one.
Third one. Okay. Now this is a tool which is freely available for everyone. Now this is kind of a software only, but it uses something called grid computer. In grid computer, what happens though? If you want to do a lot of calculation, then what would happen is, because we will be using our small computers, no, so it is not very feasible. Your computer will jam because it needs a lot of space, as well as the whole database is very huge. So what these people have done is, they have made it into a remote server. Through this server, you can communicate to the mainframe computer of Harvard University. So Harvard is having the database. So the tool is also developed by them. Okay, now what we'll do is, we'll take our data set and we'll use this particular tool to deploy our results. Okay, so Gaurav sir, as you have said rightly, we'll copy the FASTA sequence from the second file, that is BRCA partial sequence.
Yes, okay. I have copied
the FASTA. You please remember. Yeah, you please remember that you have to copy it from the greater than sign. Okay, do not miss the greater than.
Yes, sir. Yes,
sir. In the second window, please paste it. Fine, sir. Now in the second window, please paste it.
Yes.
Second
SNP
window.
identifier. This one.
FASTA.
No. Second window. Here.
Protein sequence in FASTA.
Okay, great. Yes. Okay. So till now, what we have done, participants, we have identified this particular gene sequence from the whole genome sequence. This will be done by whom? That lal path and all such things I have given. No, a quick disclaimer: I am not promoting anyone because they are already the biggest player in the market. I am just giving them examples. So standard disclaimer. Now what has happened? What these people will do? They will sequence your BRCA1, then reverse translate it into a protein sequence. Then they will give it to you. Now it is your job to find out whether you are having a mutation or not. So consider that you are having a mutation at the 6th amino acid. Okay. So what we do, sir, Gaurav sir, we will put in the position window, we will put 6, digit 6. You take your cursor to the position window, then put the digit 6. Great. Now here sequence we will see what is the 6th amino acid.
Sir, why position 6?
1, 2, 3, 4, 5, 6.
Methionine.
6. I'm just coming, sir. I'm just coming.
Okay.
Methionine. Great. So AA1, you select methionine. AA1, there are substitutions AA1 and AA2. In AA1, we will select M, methionine. It starts with an A, R, N, L, K, M. I can see the M. Gaurav sir, please select the M digit in the substitution position AA1. M लेटर पीछे 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 थोड़ा पीछे जाइए। ये तो एलानिन हो गया। चलते जाइए हाँ सिलेक्ट कर लीजिए। M सिलेक्ट कर लीजिए सर। M को सिलेक्ट कर लीजिए। नो M को सिलेक्ट कर लीजिए।
M, this one. Okay.
हाँ okay. So methionine has been selected. Why? Because I'll give you the story. Methionine will be replaced by another amino acid called aspartic acid D. Sir, AA2 में D में जाइए sir. AA2D, aspartic acid D. Okay. So what is methionine has been replaced with aspartic acid. So consider this. Methionine is a sulfur-containing amino acid which is aromatic amino acid. Aspartic acid, on the contrary, is a highly positively charged polar amino acid, which is usually very, very hydrophilic. Okay, fine. Now we'll submit the query. This is just a background. Sir, please submit the query. Submit query. One button is there. Okay. Submit query.
Submit.
Okay. Now, once you submit the query, you are entering the grid gateway interface of Harvard. Can you see? So what is the grid gateway interface? It is the interface which is connecting you to the grid computer, mainframe computer of the Harvard University. So it is running. Load is light because nobody else is working on it. Some of you are doing it. Sir, now refresh, sir. The result will come. Refresh button, sir. Down में refresh button है देखिए. नहीं यहाँ पे क्या है सर? The page that you are looking use information. हाँ यहाँ पे refresh कीजिए. हाँ आ गया.
It is completed.
सर अभी result के अंदर देखिए आप view completed, sir. Now you go to view results. Now what you did, you used the Harvard University mainframe computer by looking at your screen in your house. Once you are having your BRCA1 sequence done. The company has also told you that you are having a mutation on the 6th amino acid, which is usually a methionine that has been replaced by aspartic acid. Now you want to know what is the outcome of this? This is the outcome. See, probably damaging with a score of 0.962 means what? And you can see also sensitivity 0.78, specificity 0.95. There is a high probability that if you are having this mutation, you will end up with a breast lump, and eventually you can also go for a breast cyst removal surgery, or it can also, if it metastasis without any detection and all, it can also harm your life. Now this is the scenario of Angelina Jolie. She was having the mutation of this. I do not know whether you know or not. She has underwent a lumpectomy, and subsequently she has been recovered till date. Now she is okay. So are you getting my point? Why we have selected amino acid 6? So this is how machine learning will help you by click of this thing. And one more thing you can say that this particular result, no, it's highly what you call precise. You may not be like, you have no doubt if somebody is having this particular thing, she will definitely have this particular anomaly in her tissue. Okay, so how do you do it? Because we are having a huge amount of database. These people are working on this for a very long period of time, and all the hospitals, all the diagnostic centers, all the companies who are doing this sequencing, this particular lab, genetics of Harvard, is connected with them. Every day they are putting their results to this database. It is maturing every day, and this is a supervised algorithm. On the basis of that algorithm, it is predicting that if somebody is having this mutation, it will definitely be higher probability that it will get devastated. Are you getting? So now, as I have now, this is how this whole machine learning, artificial intelligence will work. Okay, and you are there to deploy these tools in such a way that you can build a business. Okay, the whole exercise is around this only, isn't it? So what are the other ways you can do? You can generate some method through which you can do this in a very cost-effective way. The whole process, identifying the effect of this mutation was zero cost. You had no cost for this. It's all free. So how it started, isn't it? Why is this free? It is free because they have replicated human intelligence into a machine. And now maintaining a machine is comparatively very, very less costly than that of maintaining a person. If you think that the person we are having in our hospital in Northeast, so it is very costly because we have not yet come up with some solution where By looking at a slide, the person will, the machine will tell me that okay, this person will live for this many days. Some people have developed, we have not yet developed. So there is a huge room. You can even do this, and you can become also a service provider of this particular thing, and eventually build a business around. Okay, so this was the this thing take home message, and the whole idea behind this particular session of mine. I do not know how you have this thing connected the dots. If you are having some queries, I will be happy if I can answer them. So again, a quick recap. In the beginning, we have spoken about that there is a huge
Should I stop here, sir?
And, yes, sir. Yes, sir. Please stop. Yes. Thank you. So in the quick recap that we started with this concept that there is a huge gap in the market. There is huge possibility that if you have some good insight, you can build very good business. And even if you develop your skills in this particular sector, there is a huge room. There is no worry that artificial intelligence will take your jobs. Not at all. Not for computer science. Not even for biological science. Okay. So and I think most of our participants are teachers themselves. They can also communicate the same to our the new generation of students, that you just need to focus on the thing you are doing, and there is absolute opportunity, huge opportunity out there. By saying so, I would conclude. Over to you, sir.
Sir, I have also learned some new in this session. So, dear participant, if you have any query, you can unmute yourself and ask to our speaker. Sir, it will be better, Surajit sir.
Sir.
It will be better that you have one whole day session on the analysis, hands-on. So I will plan for you, sir.
Yes, sir. Absolutely. No problem. No problem. Absolutely, sir. It will be great. We can easily.
Okay. Any query, participant?
Yes, absolutely. No, that is fine, but ask something.
Yes. So, participant, if you have any query, you can unmute.
Yes. Anything
I
you
think,
want
sir,
to
there
know,
is no
or
query.
you have some plan, we can also collaborate. If you have some plan to collaborate and all, that also you can communicate. Okay. If somebody is from computer science, if some other thing,
because sir, I also have some interest in Silco. So
okay, great.
Yes. And this is very actually the emerging of the pharmacogenomics era can help this session. The session will help to learn new things for the assessment of pharmacogenomics.
Definitely, sir.
Yes, sir. So kindly accept a moment of gratitude, sir, on behalf of organizing committee.
Thank you, sir. Thanks a lot.
I would like to felicitate you with the e-memento.
Yes, sir.
Thank you, sir, for accepting our invitation and delivering such a wonderful and hands-on session.
Yes, sir. Thank you.
Thank you so much, sir.
Thank you. Thanks a lot, sir. Thank you. Thanks. My email ID even. Email ID is okay. This is the email ID. If you can see it. Okay. Thanks a lot, Dr. Gaurav.
Yes,
I hope that your program is fruitful, and I also sincerely hope that I have added some value to your participants.
Yes,
thanks a lot.
Yes, sir. That was very really helpful, sir. So, dear participants, we are moving ahead for our third session. We have our speaker, Vivek sir, with us. So, Vivek sir is a highly accomplished expert in medical device regulatory affairs and quality management, bringing over 15 years of experience in the medical device industry. As the founder and director of TACIT MEDTEK Private Limited, Vivek sir has established himself as a trusted leader in guiding businesses through the complex landscape of medical device regulation and quality standards. So he is a qualified lead auditor for ISO 9001 and demonstrating his deep understanding of quality management system. He has successfully assisted numerous companies in registering their product in market worldwide, including India, the USA, Europe, Gulf countries, Russia, African, Asian, and Latin American countries. With a strong focus on supporting medical device startups, Vivek sir has provided end-to-end documentation consulting encompassing design and development, risk management, software verification and validation, clinical evaluation, testing strategy, and preparation of plant master file and device master file. His guidance has empowered this company to navigate audits with the confidence, obtain ISO certification, and obtain the manufacturing licenses in India. As an active contributor to the industry, he is engaged in training services and delivers sessions on various medical device subjects such as clinical evaluation, clinical investigation, their usability, design control, risk management, electrical medical device, as well as SMD. Throughout his career, Vivek sir has successfully handled a wide range of medical devices, including implantable, active, software, as well as medical devices. His expertise spans multiple medical specialties such as orthopedic, ophthalmic, dermatology, gynecology, rehabilitation, cardiovascular, dental, hospital disposable, radiation emitting products, as well as emergency situation products. So we are very grateful to have the Vivek sir as a keynote speaker in this session, the 4-week entrepreneurship development program. So over to you, sir. Thank you, sir, for accepting our invitation, sir.
Good afternoon. Thank you for the beautiful introduction. I'm really delighted to be the part of this program by Galgotia University. And today I'm going to share whatever the learnings that I have, that I have learned through entire working in the various industries and consulting to the various companies, right? Start, from the basic startup companies to the multiple countries, companies having presence in the multiple countries. So I'm going to share my screen.
Yes, sir, you can share.
So can you share my, can you see my screen, sir?
Yes, sir. Yes, sir. It is visible. Perfect, sir.
Okay. So, today's topic is the regulatory requirements for bio startups. As we know, this bio startup include a lot of companies, type of companies which are working in the healthcare sector. Startup companies are developing pharmaceutical product, medical devices, nutraceutical, homeopathic, Ayurvedic, a lot of kind of product they are developing, biological product. So these are companies called bio startups. So today I'm going to share what are the regulatory requirements. And when I say regulatory requirements means that what are the requirements that are in place to take these products in the market and to get the approvals to start selling the devices. So my name is Vivek Singh. I am founder and director of TACIT MedTech Private Limited. So TACIT MedTech is a specialized regulatory and quality consulting firm and help the companies to take their product into the India or across the world where they want to launch their product. So I help the companies to meet the regulatory requirement of the particular country and get the market clearance. So a brief about myself. I'm MTech in quality management and M.Pharm in regulatory affairs. I'm ISO 13485 certified lead auditor and ISO 9001 certified lead auditors. ISO 13485 is the medical device quality management system. So I help companies to establish a proper quality system in the companies and assess whether they are meeting the requirements laid down by the standard or not. In my career, I have worked almost all the leading countries and across the globe, which also include products in India, USA, Europe, Latin American countries, Africa, Gulf, CIS countries, and Asian countries. I have handled the product starting from the class 1 up to class 3. So means a low risk device to the highest risk involving devices. And the type of medical devices that I have handled is implantable medical devices, active or electrical medical devices, software as medical devices, and software in medical devices. I'm also a trainer on the various topics of the medical devices. For example, CE marking, US FDA filing, usability engine, clinical trial, electrical medical devices, meeting the requirement of IEC 60601. MDSAP is medical device single audit program. So this is a brief about my qualifications. So in presentation, what I'm going to discuss is that a general idea about what is our regulatory affairs and why it is very important for all the companies or startup companies who are working in the bio startups domain or medical device domain, why regulatory affair is very important for them. And since we are based in India, so I will be talking about the regulatory authority which you need to deal with and the regulations, the medical device regulation of the India. Then we will briefly discuss about what kind of products are the medical device, what product which look like a medical device but not a medical device, and how you will classify your medical device. And then we will be talking about the startups working with the medical devices and general regulatory strategy for the startups. So what is the regulatory affairs and why it is very important? Okay, so regulatory affairs is the interface between the regulatory authority and healthcare product manufacturer. So there is a company and the company needs the various kind of licenses from the regulatory authority. Okay, and without licenses, they are not able to launch their product to the market. So usually the companies establish a department called regulatory affairs, and the purpose of this department is to take care of all the regulatory requirement. And when we say regulatory requirement, it means these are the mandatory requirements. Failing to meet these requirements, licenses, closure of business, penalties, penal action, so just a second. Okay. So it is very much important. So let's understand what is the regulatory authority. So regulatory authority, these are the government agencies responsible to enforce the laws and regulation of the country and to ensure that only safe and effective product comes in the market. So a regulatory authority is authority established by the our government. So in our case, there is central government and the various state governments. So central government has established a regulatory authority, state government has also established various regulatory authorities. And through these authorities, our government enforces the regulation of our country for the products. And the purpose of doing is, just to make sure that only safe and effective products comes in the market. Now, if we talk about the healthcare products, so healthcare products like pharmaceutical product, medical devices, in vitro diagnostic, biological product, nutraceutical product, and these are very different kind of product. A pharmaceutical product is like tablet, capsule, or injections. Medical devices, something looks like a machine or software or some kind of equipment which you generally see in the hospital, sometime also as at home. In vitro diagnostic, it's like some kind of machines or reagents which we use to identify a disease. Biological products, for example, vaccines. So these are the example of biological product, nutraceutical products. So these are the different kind of healthcare products. And before you, if you are in the business of these kind of product, then you need to follow certain regulations and you need to understand the regulatory requirements. Without understanding the regulatory requirement, one will not be able to take these products in the market. Now we'll talk about the bio startups. So bio startups, it's like the startup working with the healthcare products. So for example, a startup working with the pharmaceutical product or medical device or IVDs, these are the startups we generally call as an bio startups. Now, just to understand the interface complexity. So I told that regulatory affairs, regulatory authority and healthcare product. So for one healthcare product, for example, pharmaceutical or medical devices or in vitro diagnostics, we have a different set of rules in India. So if there are, let's say, two startup companies, one is working with the pharmaceutical and one is working with the medical devices, both of the companies have to deal with the different kind of regulation. Similarly, if a company working in the medical devices in India, and if they want to take their product to the Europe, then again, there are different set of rules for if they want to work in India and they want to take the product to the Europe or any other country where they want to sell the product. So it increases a lot of complexity. So which regulation that you need to follow if you're working in India, and what sort of regulation that you need to follow if you want to take the product outside India and want to export your product. So it is very important. Ensuring compliance with the regulation is crucial, as failure to do so can have serious consequences, including potential harm and imposition of penalty by regulatory authorities. So as I told you, for example, there are certain rules even for the traffic management. If there is a red light, you can't cross. If you cross the road, if you jump the red light, then there are some penal actions. Similarly, there are certain rules and regulations for the pharmaceutical, medical device, in vitro diagnostic, biological product, every kind of products for these healthcare product industries. And if you will not follow those regulations, then there could be a penal action and by the regulatory authorities. So for this presentation, I'm going to talk about the regulatory authority. A focus of this presentation will be regulatory authority India, and the healthcare product I'm going to talk about is the medical devices. So for medical devices, what are the applicable regulatory requirement in India? We will be discussing in this presentation. So Indian Regulatory Authority and Medical Device Regulation, the various stakeholders. So as I told you, there is a central government and there are state governments. So central government has one regulatory authority and it is called CDSCO, Central Drug Standard Control Organization. Their headquarters is in Delhi. And the state regulatory authority, it's Food and Drug Administration of different states. So for example, if your company is based in Maharashtra, then sometimes you may have to deal with the Maharashtra FDA. If your company is based in UP, then you may have to deal with the Uttar Pradesh Food and Drug Administration. So these are the state authorities. So one is the central authority, another is the state authority, and these two are government authorities. There is a third term called notified body. So notified body means a body, corporate or other legal entity registered under Rule 13 as a body competent to carry out the audit of manufacturing site, assessment and verification of specific categories of medical devices for establishing conformity with standard. So notified bodies are simply private companies, okay? And these private companies are licensed by the government authorities like CDSCO to perform audit of the manufacturers. So in general, audits are being performed by the government authorities. But here, in this case, government authorities has given licenses to some of the private companies to do audit on their behalf and submit their audit report. And example of such companies are like TUE, DNV, BSCIC. So these are some companies which are certified by CDSO to audit the medical device companies. So what is the act and rules that we need to follow if we are working in India? So one act is the Drug and Cosmetic Act 1940 and Rules 1945. This is the major act which everybody needs to follow working in the pharmaceutical, medical device, biological, all kind of products. However, there is a specific medical device rules 2017 which was drawn by the central government. And if a company is a medical device company, then the rules applicable for the medical device, medical rules given in the medical device rule 2017 is applicable to these companies. So if you are a medical device company, for you, medical device regulation 2017 is the go-to regulation. So now, as a company, whom do you have to deal with? Okay, so as I told you, there are various stakeholders, central government, state government, notified bodies. So if you are a medical device company, whom do you have to deal with? So it depends on the risk class of your device. Higher the risk to patient or user, higher the risk class. So in India, the Indian government has classified medical devices into 4 risk classes. These classes are A, B, C, D. Okay, so here in this picture, I have shown the medical devices in class A, B, C, and D, and these are the regulatory requirement. You can also say that these are the risks to the patient and user. So if a device is class A, then the regulatory requirements are low and risk to the patient is low. If the device is class D, then regulatory requirements are higher and risk to the patient is higher. So if you are working with the class A and B medical devices, means low risk medical devices, then you have to deal with the state FDA and notified bodies. But if your product is a high risk product, then you have to deal with the Central Drug Standard Control Organization, CDSCO, that's central government agency. So I hope this is clear. For low risk devices, the enforcement is given to the state FDA, and for the high risk devices, enforcement is given to the central government. So what is a medical device? So in general, medical devices are some very common machine equipment we see in hospitals or at home. Okay, and generally we use such kind of product for the diagnosis and treatment purpose. So without a medical device, even routine medical procedure from bandaging a sprained ankle or implanting an artificial hip or any surgical intervention would not be possible. So medical devices, even you can see at home and some high-end devices in very specialized hospitals. So these are variety of medical devices. Now, which are the users of the medical devices? So in some cases, a user could be a lay person. For example, we take the thermometer at home and we measure our body temperature. So we are not a very trained medical person, but we can use such kind of medical device like thermometer at home. So even a lay person can use a medical device at home. Some paramedical staff or clinician in the remote clinic, like for example, taking blood pressure, using a blood pressure machine, or a highly trained healthcare professional in advanced medical facilities who are using MRI machines, robotic robotic arms to perform the surgeries, such kind of high-tech medical devices. So variety of people from layperson to paramedical staff to highly skilled doctors, everybody uses the medical devices. And the purpose of the medical device is to diagnose an illness, to monitor a treatment, to assist disabled person, or to intervene or treat illness, both acute or chronic. So medical devices help in diagnosis or treatment of any kind of diseases. And this is not the very latest estimate, but in general, there are more than 5 lakh different kind of medical devices on the world market, separated into more than 10,000 generic device groups. So there are huge diversity in the medical devices. So some medical devices could be like an intraocular lens, which is going to be implanted in the eye. Some medical devices could be a pacemaker, which is going to be implanted inside the body on the heart. Some medical devices are just bandages we are using on our intact skin, scissors. So these are very different kind of medical product, and all are having same kind of regulation. So, how to take these products into the market is a very critical job. Now let's try to understand how this medical devices looks like. Okay, so I have given some pictures over here. So let's see. So this is the first product called bone plate. So, in case of fractures, sometimes doctor do a surgery and put the plates and tight up these plate with the help of his screws. So in the below image, you can see it's X-ray, So in X-ray, you can see that how these plates are implanted inside the body and how these are screwed up. So doctors use such kind of product. These are implantable products. This is a medical device. Very common stethoscope. We have seen this from our childhood days. So stethoscope, also a medical device. Hip implants, a very high risk device. In case of severe accidents where this femur bone get fractured and the person get permanent disability, this hip implant help the person to regain, the movement. So this is a high risk device. Digital thermometer, everybody, we have seen this kind of product at our home to measure the body temperature. MRI machine, very highly skilled high-tech device. MRI machines, it's a medical device. X-ray machine, it's a medical device. Pacemaker, again, it's an implantable device. These wires are, this is used if some people having abnormal heart rhythms. So this product is used to normalize the heart rhythm so that heart functions properly. This implanted inside the body, and this wire goes and touch the various lobes of the heart. So this is a medical device. This is external fixator. So you can see that this person's leg, it has multiple fractures, and it's not possible to keep all the bones same in their position just by putting a plaster or doing a surgery. So you can see that the multiple screws are used, and these goes inside the body to keep the product in the bones at their location. And these screws are then tightened up with the help of this external fixator. Again, this product is a medical device. Very common, very simple product, scissor, which is used in the surgery. It's a medical device. Now here is a surprise. So, eye drops. So is it eye drop? Is it possible that eye drop could be a medical device? Yes, it's possible. So we'll see this once we will be talking about the medical device definition, then we'll see how an eye drop could be a medical device. Drug-eluting stents, very popular these days because of heart blockage. Many times people detected with the blockage in their coronary arteries. So this product is used to open up the arteries and resumes the blood circulation over there. So drug-eluting stent. Surgical gloves, syringes, again, medical devices. Refilled syringes, medical devices. This is an operation room where surgical light, bed, robotic arm, monitoring machines, all these products are medical devices. Blood pressure monitoring machine, softwares, self-HIV testing kit, condoms, blood bag, ventilator machine. So during Corona, we saw this ventilator machines become very popular. So these products are also medical devices. So with these pictures, I wanted to show you that these products varies a lot. Even a software can be a medical device. An eye drop could be a medical device. Blood bag, condoms, and very high sophisticated machines are medical devices. So let's understand the definition of the medical devices. It's a long one, but I'll summarize this for you. So I'll read it once and then I'll explain the definition. So as per GSTF, GSTF is the Global Harmonization Task Force, and they first came and they gave the definition of medical devices, which is in general acceptable globally, even India having a similar medical device definition. So as per GSTF, medical device means any instrument, apparatus, implement, machine, appliance, implant, reagent for in vitro use, software, material, or other similar or related article. So if you can see till here, instrument, apparatus, implement. So they are talking about various kind of products. Okay. Now, these products intended by the manufacturer to be used alone or in combination. Okay. So these products either can be used alone or in combination, and most important, for human beings. So all these products has to be used for the human beings for one of the following specific medical purpose. So if you are having an instrument, apparatus, implement, machine, and it is going to be used for the humans, and if your product is going to do one of these following medical purpose, where what are these medical purpose? Diagnosis, prevention, monitoring, treatment, or alleviation of disease. Diagnosis, monitoring, treatment, alleviation, or compensation of an injury. Investigation, replacement, modification, or support of the anatomy or of the physiological process. Supporting or sustaining life. Control of conception. Disinfection of medical devices. Providing information by means of in vitro examination of a specimen derived from the human body. So this is long definition. So I was talking about the medical purpose. So these are the medical purpose. So if you are making a product, okay, for the human use, and if your product is doing one of these following activities: disinfection of medical device, control of conception, supporting life or sustaining life, if doing such kind of things and does not achieve its primary intended action. So this is very important. Primary intended action by pharmacological, immunological, or metabological means in or on the human body, but which may be assisted in its intended function by such means. So product, then its medical use, and how it functions. So it should not achieve its function by pharmacological means, because if it is doing achieving its function by pharmacological means, then it will become a pharmaceutical product. If it is achieving its above the medical needs by doing an immunological action or metabological means, then it may be a biological product or again a pharmaceutical product. So medical device will also treat a disease, but it will not have a pharmacological, immunological, or metabological action on the body. Okay, I hope a little bit you have got the clarity. So this is very important. Typically, the medical device function is achieved by physical means, including mechanical action, physical barrier, replacement, or support to the organ or body function. Okay, so just to explain this point, I am going to a product image. So here you see, so this is a bone fracture, and these implants are, after the surgery placed on the bone, and screws are tied up here. So basically this implant is not healing the bone. It is the bone itself, the bone heal itself. Okay. But this implant, what the implant is doing, they are keeping the bone in immovable position so that bone can heal itself. So product is not having any pharmacological, immunological, or metabological action on the body, but it is treating a condition, and the fracture condition. Okay, so because of this bone plate is a medical device. I hope now a medical device definition is a little bit more clear to you. Similarly, a medical device may be supported in its function by pharmacological, immunological, or metabological means, but if these becomes the primary mode of action, the product is no longer a medical device, but instead it is regulated as a medicine. So as I explained you that even the pharmaceutical product also treat the disease or having a medical purpose, but if the mode of action of the product is through a pharmacological, immunological, or metabological means, then that product will not be medical device, maybe a pharmaceutical product or a biological product. So again, just for more clarity, what product may not be a medical device, which look like a medical device but may not be a medical device. So this is a coronary stent. Okay, and I have shown you the picture of this product, and this is a drug-eluting coronary stent. So there, this is a metal body, and this body is coated with the drug. Okay, it is having a polymer coating of the drug in it. So this is a drug-eluting stent, and it comprises of medical device and drug. Now the question is it a medical device or a drug? Because now a device and drug is combined together. Okay, so how it will it be regulated? So if you make such kind of product, any startup makes such kind of product where they integrate a medical device with a biological product or a medical device with a pharmaceutical product. So will your product be a medical device or pharmaceutical or biological product in that case? So let's understand. So a new category of product has come up, and this is called the combination product. And don't get confused with the fixed dose combination where we combine two type of drugs in one tablet. Okay, so this is a drug, this is a medical device, this is a biological product. Okay, and when you combine the drug with a medical device, it becomes a drug medical device combination product. And if you combine the drug with the biologics, then drug and biologics combination product. And if you combine the medical device with the biologics, so medical device and biologics combination product. So you can see that these are very different kind of product, regulated differently, entirely their regulation is different. Okay, you have to generate very different kind of data to take these products on the market. They are having different kind of mechanism of action when they have to treat a disease or a diagnosis disease. So these products are entirely different, but when you combine these products together, then you make a product called combination product. Now, is a combination product a drug or medical device or biological product? So it depends. It depends on what is the primary mode of action of the product. So if you combine drug and medical device, obviously both product will have some action on the body. Okay, some action will be a primary mode of action of the combined product, and one action will be a secondary mode of action. So let's understand this by some examples. So a combination product has differently regulated products such as drug, medical device, or biological products. Now question, a combination product is having drug and medical device. Whether the combination product will be regulated as medical device or drug, how will we determine that? Okay, so I showed you one image of drug eluting stent where drug is coated on a medical device. Okay, so how it will be regulated? So it depends on the primary mode of action. The purpose of drug eluting stent is to open the blockage, blood blockage in a vein. So when the doctor does the surgery, put a stent inside the blood artery and open up the artery, and the flow resumes. So this is the primary mode of action of the combined product, is to remove the blockage, and it is coming from your medical device part. And because of that reason, this product may have the is having the drug, but overall product is a medical device. Okay, and the drug part over there, the coating is to just to stop the cell division. During at that sometimes at during surgery, some scar happen inside that artery, and because of that, very rapid cell division starts over there. So just to stop that kind of cell division, the drug is coated on the drug eluting stent. So drug is not actually resolving your blood clot problem. It is the device part which is resolving the drug clot problem. So because of that, the combination product is called as a majorly regulated as a medical device. The second example is the drug eluting disc to brain cancer. So there is a disc, and this disc is coated with a drug, and that goes inside the brain to treat some kind of tumors. So here the treatment is not coming from the disc part, it is coming from your drug part. So because again this is a drug eluting disc, so this disc is primarily a drug, not a medical device. So in this way you have to identify what is the primary mode of action of the combination product, and based on that you can determine if the product is medical device or not a medical device. So I was talking about some product may look like a medical device but may not be a medical device. For example, your drug eluting disc, which may look like a medical device but it's actually drug. So here I have given some example. Drug device combination product is like drug eluting coronary stent, syringe filled with the drug, biologic device, syringe filled with a biologic, and drug biologic chemotherapy therapeutic drugs combined with a monoclonal antibodies. So till now, I was discussing about the medical devices, what is a medical device, what product look like a medical device, and what product may not be medical device. Now, once you get confirmed that your product is a medical device, so one of the early thing that you need to do is that you need to determine the medical device class, risk class of your medical device. And how you need to do that? So medical device classification, you need to determine for your product. One of the first step in your regulatory journey is to determine the risk class. And based on the risk class of the device, we can determine the data and effort, infrastructure and finances that you need to take, the product to the market. So if during the medical device classification earlier, one of the slide I told you that A and B will be regulated by state FDA, C and D will be regulated by the central government. So if your A and B is a low risk device, C and D is a high risk device. So if your product is low risk, then usually you need less data and you have the less regulatory requirement in comparison to a product which is a high risk. So in case of high risk devices, the higher data requirement and higher regulatory requirements are in place. So now let's assume you have a product, it's a medical device, and you want to classify. So first you need to determine if your product is a medical device or not. So you need to check these three conditions if you're meeting these three conditions or not. First, your product should be intended for human use. Okay, so if your product is not for the human use, it will not be a medical device. Secondly, it's intended by the manufacturer, its labeling or IFU, at least one of the medical purpose listed in the definition of the medical device. It means that your product should be for the monitoring, treatment, or alleviation of the disease. I was telling you the various, definition, telling you the definition of the medical device, where monitoring, measurement, treatment, conception, life support. So such kind of purpose, your device should be having such kind of purpose. So first, it should for the human use, and it should have a medical purpose. And third, the primary mode of action must not be metabological, immunological, and pharmacological. If your product is meeting these three criteria, then your product is a medical device. Now you need to determine if it is A, B, C, D. Okay, so again, determining ABCDs is a complex process, but the various factors which plays role in determining the risk class, it's like intended use, duration of use, kind of contact, or active or not active. Means if you make a medical device which is going to be used on your central circulatory system or nervous system, in that case, your product may be very simple, but its risk class will be higher. Duration of use. If you're making a product which is coming in contact just for a few seconds, and if you're making a product which goes inside the body and remain there forever. So if the product goes inside the body and remain forever, so that will be again a high risk device. Again, the kind of contact. If your product is just touching the bare skin, or it is going inside the body, if it is invasive or non-invasive. So if the product is invasive, the risk class will be higher. And active or not active, it means that if your product does it require any kind of electrical supply or power source to work? If yes, then risk class is higher. So these are some factors which increases or decreases the risk class. So giving this in more detail, if your product is for the life supporting or sustaining, risk class is higher. If your product is invasive, okay, so how invasive is that? So, if it is up to what extent and for how long, if it is there, then risk class is higher. Sometimes your medical device may include a medicinal product. So if it is there, some medicinal products are, combined with your medical device, in that case, the risk class will be higher. Again, if your medical device incorporates a human or animal tissue, like a biological product, then again, the risk class will be higher. Is electrically or pneumatically powered. So if it is electrically powered, risk class will be higher. Delivers medicinal product, energy or radiation. If you're making a product which is intended to deliver energy or radiation, like gamma radiations and X-rays, and or if it is for the delivery of the medicinal product, risk class is higher. And if your product could modify blood or other body fluid, risk class is higher. Again, if you make a device and you say that this product is going to be used at the home by layperson, okay, then in that case, risk class will be higher. And if it is used in combination with other medical devices, again, the risk class will be higher. So there are various factors which affects the risk classification of the medical devices. And in this example, I've shown you this is a class 1, class A, class B, class C, and class D devices. And from class A, you if go up to class D, then you can see the regulatory requirements are increasing. Okay, and if some of examples of class A devices are like band-aid, surgical mask and gloves, stethoscope for diagnosis, device intended for general examination, for external patient support, for example, hospital bed, walking aid, wheelchair. So if you see, if these kind products get failed, then it's not like your patient is going to die. Okay, but in case of class D devices, if these products fail, patient may get die, may have permanent disability. It may require surgical intervention to to treat the patient if the product fails. For example, heart valves, bone implants with antibiotics, coronary drug-eluting stent, hip implants, spinal needle. These are very high-risk products. So failure of these products would have severe consequences on the patient. And B and C are, in the moderate range. B is some example of orthodontic wires, external fixators, needle for syringes, and class C are bone plate, bone screw, hemodialysis lines and machines, ECG ventilator, ultrasound, fatal heart detector. So such kind of machines are class C devices. So till now we have understood what is the regulatory affairs, what is regulatory authority, what is applicable regulation for India, medical device regulation 2017, what is a medical device and what is not a medical device, and medical device risk classification. Now, this is about the the basic that I wanted to discuss with you about the medical devices. Now, if you are a startup company and working with the medical devices, then what should be the way forward? What should be the correct steps that you need to take? So first is that you need to determine if your product is a medical device or not. I've already explained the way how you can determine if your product is medical device or not. Secondly, if your product is medical device, determine the intended use. And this is very important, what kind of claims that you are going to make with your product. So this is a 1 line, 2 line, 3 line statement, but this 1 or 2 or 3 line statement can really impact the overall process of taking your product to the market. If you have very specific claims about your medical device, then then your product may go into higher class. You may need higher funding, higher infrastructure, and more timelines to take the product in the market. But if your product is not having a very stringent or very strong claims, then the product could be in low risk category and you can easily take that product into the market. Then the third step is to determine the risk class of your device. If your device is class A, B, C, D, okay, then prepare a regulatory strategy to take the product in the market. Then after all these steps, now you confirm that your device, you are working with a medical device, this is the intended use, this is the risk class. Now you need to make a proper regulatory strategy, a sound regulatory strategy. What is the way forward from here up to the final market launch, and follow your regulatory strategy and update the strategy as and when necessary. So I have shown this in form of some simple, in a simplified way. Is steps to move forward. Is your product a medical device? Yes. What is the intended use? Determine the intended use. Determine the risk class. Prepare the regulatory strategy. Follow your regulatory strategy and amend the strategy as per the need during the regulatory process. And I'm going to talk about a very general regulatory strategy for medical devices. This is a very general way because many medical devices are at different development stages, and they are maybe at the different life cycle of the medical devices. So this strategy may not be 100% applicable to them, but it will give an idea for a medical device startup who are still in the very early stages of their design development. They can follow this kind of a strategy. So if you're if you're ready with your idea or conception, then the first step that you need to do is start implementation of ISO 13485. Okay, and ISO 13485 is the medical device quality management system. And this system, this is one of the requirement even in India that you need to take care of. So in India, medical device rule, Indian government has laid down the regulation, which is very similar to ISO 13485. So if you implement ISO 13485, it means that you are meeting the Indian government requirement. So at the time of audit, the auditors will come to your factory and they will see that if ISO 13485 is implemented or not. During your design development process, have you considered the regulations or provisions written in the ISO 13485 or not? And if you are in the hurry, then what you can do is that first implement the necessary processes like management process, design and risk. So these are some preliminary and important processes which you need to implement. So, and then keep on implementing other processes, parallelly start the design documentation and risk documentation. Okay. So being a startup company, many type companies, they want to come to the market as quickly as possible. So if you see that ISO 13485 implementation, it may take somewhere around 3 month to 6 month timeline. So do we have to wait up to that timeline first to finish the ISO 13485, then start developing the medical device? Answer is no. First, implement some important processes. For example, you are going to develop your medical device. So they implement the process for the medical device development, okay, design development. And during the design development, you also need to identify various kind of risk associated with your medical device. Then also implement this process. So some 2, 3, 4 processes which are very critical in nature, then first implement them. And then you start doing your design development activity and start developing, implementing other processes. For example, customer communication. So if you are a very beginning stage, you are not starting customer communication immediately. So you can implement it later, but before you get an audit from an auditor, before that all systems in a place. But to expedite the your regulatory process, you can implement some processes earlier and some processes in parallel with your design development activities. And here in blue color, I have mentioned the various kind of costing that you may have to incur during the this whole journey. So ISO 13485 implementation. Sometimes if you are a new company and this process is a little bit complex, then you need help of some consulting firms to help you to implement ISO 13485. So here a consulting cost comes in. Then when your ISO certification, you are ready for the certification, then you contact an notified body, and that notified bodies will come then for your audit and issue your certificate. So they will charge you, notified body charges will be there for ISO 13485 certification. Once you have ISO 13485 certification, then what you can do is you can apply to CDSCO for test license. And what is a test license? Test license is a license to manufacture your device in small quantities for testing and analysis purpose. Okay, so apply for the test license. Mention that you want 10, 20, 30, 50 pieces you want to manufacture for this test, and you will get the approval from the CDSCO. Once you have the approval, perform those tests, generate the testing data, compile the data, and then submit the dossier. So at the time of test licensing application, the cost could be, for example, consulting cost, government fees, testing lab charges. You may have to perform clinical trials as well. So clinical trial charges. So that kind of charges will trigger at this stage. And now once you complete all this testing analysis, then you will compile the dossier for the manufacturing license and submit. Again, it's a complex process. Initially you may need help of some consulting firms. So consulting cost will come into the picture. Then government fees, notified body charges. So if your product is a class A and B device, then notified body will come for an audit. So their charges you may have to bear. For high risk devices, the government fees is really high in that case, and notified body charges may not be applicable in that case. So after submission of the dossier to the authorities, they will review your whole data, what kind of test data that you have generated, what kind of claims you are making, how your device is safe, is your device is quality product, if your device is really effective. They will verify the complete data. They will come back with the queries to you. Also, or you will receive an audit at your factory, either by a notified body or from the government agencies like this medical device inspectors from the state and the center. They will come, they will inspect your facility for compliance, especially with the ISO 13485, and they will also verify if you have generated the data, whether that data is genuine or not, how you have generated the data. They will verify all these things. And once these all process is over, then you will receive the manufacturing license. And the manufacturing license is the approval from the government that now you can start selling your device to the general public. So this is a general regulatory strategy. To simplify this, first step, implement ISO 13485, receive audit and get ISO 1 certificate. So once you apply, you may get an ISO 13485. After audit, you will get an ISO 13485 certificate. Then apply for the test license, complete the product testing, compile the dossier and apply for manufacturing license, face audits, respond to the queries, and finally you will get an approval of manufacturing license. So during this whole presentation, we talked about various things. So what are the key takeaways that we need to consider? First, the step is to determine if your product is medical device or not. Second, determine the risk class of your device. Consider the various factors discussed in this presentation to determine the risk class. If your product is A, B, C, D, determine it, okay? So this is very much important that you have to understand that medical device is a regulated industry, and regulatory compliance is very, very, very critical. So not just for medical devices, all healthcare products. For example, if you're in the pharmaceuticals, if you're developing the biological product, if you are working with the similar kind of healthcare product, combination product. So regulatory, so you are in a regulated industry, and it means that the laid down regulation are very much critical to you, and failure can lead to legal and penal action, sometimes even jail or closure of the high penalties, and sometimes shutdown of the factories as well. Okay, so what you should do in that case? So involve the regulatory professional. Regulatory professional should be involved right from the starting of the design and development phase of the devices. Failure to do so may trigger the redesign of the device. So if you are developing a device, involve the regulatory person who is competent enough to understand the kind of design requirement for your devices. Okay, so if he is involved right from the beginning of your design process, he can give you various input, which is very critical for the product to come into the market. Otherwise, you may develop a device and then you go for, you say that now I'm ready for the submission, and then you come to know that your design is not meeting certain provisions. So then you may have to redesign your product, which can ultimately lead you, redesign the process, which can delay your market launch, and again, putting extra effort for the redesigning of the medical devices. So regulatory professional involvement, right, starting from the design development is very much critical. Okay, each device is unique. Various manufacturers' device could be at various stages of the device life cycle. Hence, a tailored regulatory strategy should be made. Also, changing regulation may impact the predefined regulatory pathway of the device. So if you are making a regulatory strategy for your product, what is the risk class, what should be the what data that you need to generate, and suppose after 1 month there is certain change in the regulation, and then that regulatory strategy you have to revisit. You again need to check what is what these new regulation are going to impact on the medical devices. So for example, some product earlier used not to be regulated. Now these products comes under the regulation. Some product earlier were on the low risk. Now CDSCO has said that they will be having a high risk classification. So those manufacturers now have to have to generate more data to take the product into the market. So you have to be very active as per when it comes to the regulatory strategy. Keep on focusing on that. Not following correct regulatory path may void all the data generated on the design on the device and may delay the scheduled launch of the device. So for example, you make a device and you perform various kind of tests on your device, and then you come to know that there was a certain license which you need to take before performing the test. So it may be possible that you have performed a test of 2 lakh, 3 lakh rupees and you do not have a license. And at the time of audit, an auditor may say that since this product is, this data is generated without having a test license, so we will not consider this test report. So, this is the kind of problem, not having a proper regulatory strategy. Again, a sound regulatory strategy may avoid some expensive tests and help the companies to take the product in market in less time with less expenses. Sometimes, you may plan that I'll be doing some clinical testing, I'll be doing some XYZ testing, and there is possibility that even without doing those tests, you can take the product to the market. There could be some alternative. Things can help you to avoid such kind of expensive test at that time. A good regulatory strategy will help the company to generate the data in such a way that can be used for registration in multiple countries. So for example, you develop a product and you want to register the product in India, but ultimately everybody wants to be global. Okay, so have a vision to go global from the beginning and generate the data in such a way that the data that you are generating in India, you could be able to use that data for the international registrations as well. Okay, so this is again a very critical thing, and regulatory strategy help the companies to bring a safe and effective medical device in the market. Okay, and help the company to establish itself as a very reputed brand. So, through this presentation, I wanted to highlight that the, regulatory requirements are very much critical, and being in a medical device, pharmaceutical, biological, or, in the healthcare sector, it is a very important for the medical device manufacturer to meet this regulatory requirement and be in compliance with the applicable regulatory requirement. This will help to take a sound, safe, and effective product to the market, and really help the company to go global. And with that, I conclude my presentation, and I'll be happy to answer the queries if you have. Thank you.
Such an exhaustive session, sir. Very informative. I have also learned several new things. So, dear participant, please unmute yourself and ask your query, if you have any. Anyone, if you want?
May I?
Yes, please.
It's as you have mentioned, it's a wonderful session, and I would like to thank the Dr. Vivek sir for that wonderful explanation. Actually, I just have to say one thing, that even before going for any startup or business, this is the very beginning thing in the beginning, that are regulatory frameworks that we are very much afraid of. So he has explained it wonderfully. So thank you so much for that explanations and all, and looking forward to have interaction with you later after this workshop as well.
Sure. We have also based in Greater Noida, so sometimes if you want, we can meet also. My office is in Greater Noida itself.
Any other query? Okay. So I think there is no query.
That's really great.
But participant is very happy. They are giving their feedback, sir, in the chat box.
Yeah, I can see that.
Yes, yes, sir. So we are very thankful to have you, sir, as a keynote speaker. So kindly accept the memento as a gratitude. And we are again thankful. We are grateful that you have shared your vision and joined our endeavor. So once again, sir, very thank you so much.
Thank you, Dr. Gaurav, and all the participants. So I really, I really want this zone, North Zone, this Delhi and North Zone area. Students really should take this medical device startup journey very seriously. Even, near Zevar Airport, this medical device park is coming up. So in India, there will be 4 medical device parks. Even somebody who is looking up a career in the medical device industry. So this is a great time.
Yes, sir, of course, sir. Sir, I will contact you, sir, and I will invite you for the physical session also.
Sure.
Yes, sir. Currently, we have planned one faculty development program on entrepreneurship.
Okay.
From that will be start from the 3rd of the August and will end on the 17th of the August. So after 17th of August, I will contact you, sir, for the session, offline session, and we really need your guidance, sir, because our student is also doing very good work. We have approximately 100 plus startups in our incubation center, and they are needing some mentors who can translate, who can help, and who can support their idea to translate into the startups. So yes, sir. So thank you,
so
sir.
much, Dr. Gaurav. And it was really pleasure talking to sharing my knowledge with you. And whenever if you have a query, you have my number, email ID.
Yes,
you can always contact me.
Yes, sir. Of course, sir. We will contact you, sir.
Somebody is asking how to may I share my number. My number is 94. Just a second, Dr. Gaurav.
Yes, sir. Yes, sir. Please.
Sharing my number so that if somebody wants to contact me, they can. Can we conclude this session now, Dr. Gaurav?
Yes, sir.
Can we conclude this session now?
Yes, sir.
Okay.
Yeah. Thank you. Thank you so much, sir.
Yeah.